Hey guys, how are we doing? Can you hear me okay? Let me know in the chat if you can hear me okay. I can see the chat popping already. Phil, hi. We got Jathana, Demipo, Minto Play, Deborah. Good to see you all. I think some of you did you guys go to the physics boot camp on Sunday? How was it? Did you guys find it useful? We're doing a similar thing today. So this is the schedule, guys, for the next 5 sessions. So we're done with physics paper 1. Let's put a tick there. We're doing bio paper 1 today. So plan is to go through literally all the content in bio paper 1 today. We're starting now. We'll be finishing around 3:30 p.m. Hi Frozen, hi TDK, hi Zohar. It was good. That's good to hear, guys. That's really good to hear. So I hope you guys have your revision guides in front of you. They're going to be really useful because that is literally what we're going through today. I'm going to go through all the points in the revision guide. I'm going to be explaining it. I'm going to show you all the memory tricks as well. Bio is going to be different to physics. There's so many memory tricks I have for bio. So it's going to be a lot of those. Physics is more calculation based, but today you're going to see there's quite a few that I'm going to be going through today. So yeah, if you guys have anyone who you think will find this interesting, please guys share, subscribe if you want to chat on the in the chat as well. Share it to your friends. Don't gatekeep. Let them know we're doing this for free. We're doing all the paper ones for free. And yeah, let's see what this chat is saying. Cell biology. That's what we're starting off with. Minto Play. Oh, was that you, Dylan? Dylan, congrats on winning the spin the wheel. Yes. So that's you. You're going to get everything for free. So that's all good. XP. Yeah, I don't know how XP works on YouTube. I honestly don't know how you get XP. Does anyone know? I don't know. So we can't reward people for the XP. Let's see. Paper 1. We're going through AQA basically. But it is very relevant for the other exam boards, OCR and EdXL as well. It's going to be relevant there. So yeah, guys, if you have any questions, drop it in the chat. If you have any like questions about the revision, guys, if you haven't got them or anything, then send an email to help@flashrevisionlab.com. All right. Let's see. Anything else for me to mention? Okay, so this hasn't happened for over a year. Our streams have never gone down for the last year, but just in case they do, stay on the screen, stay on the on this particular stream, and we'll try and reconnect. If that still doesn't work, we'll send you guys an email. So keep an eye out on your emails. But fingers crossed that won't happen. It hasn't happened to us in a very long time, so it should be fine. In terms of content, we're going to be mainly following the combined syllabus. And if you do triple, those of you who do triple, we'll be doing some extra sections just before the end of a session. So the session is going to look like this. So we've got 4 sessions. And just before break, if you guys do triple, we'll be covering the triple content here. So those of you who do triple, there is a bit more for you guys to do. And you're going to get slightly smaller breaks, but hopefully it'll be worth it in the end as well. If you do foundation, then you will have to look at the revision guide that we sent out. In the revision guide, we've got the spec section. And in the spec section, there's sections that say HT only or higher only. If it says HT only or higher only and you do foundation, it means you don't need to know that topic. So how can you get physics paper 1? You can still get it, I think, if you just sign up on the same website again. You can still get physics paper 1. So guys, a lot to get through today. So keep asking me questions in the chat. I'll do my best to respond. We've got 77 people here right now, so I'll do my best to respond to everyone. But yeah, let's get started. The first topic we're going through is cell biology. And we're going to go straight into a memory trick, guys. We're talking about eukaryotes and prokaryotes. If you guys know the memory tricks because you've watched my TikToks, then let me know in the chat as well. That's always good to see as well. We're talking about the difference between eukaryotes and prokaryotes. So look what I've done here. I've written the words eukaryotic and prokaryotic. and I'm going to use a memory trick to remind you what each of these are. So I've purposely written eukaryotic with a capital E and U. Does anyone know why? Prokaryotic. I haven't done anything to prokaryotic. It's just written as normal. EU in eukaryotic is because eukaryotic cells have a nucleus, and nucleus has an EU in it. Oh, that's not where the that's not where the EU is. Nucleus. Let's make that small s. So you guys can see eukaryotic has EU in it. Nucleus has also got EU in it. So that can help you remember that eukaryotic cells have a nucleus in it. Prokaryotic, pro rhymes with no. No, it does not have a nucleus. So we can say here it has no nucleus. Why do I keep writing it like that? Nucleus. Another thing you can say, eukaryotic starts with U, as in you are human. In other words, animals. So eukaryotic organisms are organisms such as animals, like you, and plants, whereas prokaryotic are the opposite. They're not animals and plants. Examples of prokaryotic cells are bacteria cells. They don't have a nucleus. Okay, so that's the first memory trick there. Yes, 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 because the EU stands for you as well. So what do we have here? Main thing, they have a nucleus. That's the main thing about eukaryotic cells. Prokaryotic cells do not have a nucleus. Eukaryotic cells are larger and more complex, whereas prokaryotic cells are usually smaller and simpler. So in terms of cells, there are 3 types of cells you guys need to be aware of: animal cells, plant cells, and bacteria cells. And for those, you need to know the labels, you need to know how to label them, and also you need to know the meaning of each label. So if we look at an animal cell, what we have on the very outside, we have the cell membrane. What does the cell membrane do? It's like the outside of a water balloon. Think of it as like a water balloon, the outside, the skin of it. It controls what goes in and out of the cell. Then we've got the cytoplasm, which is the water inside the water balloon. It's a jelly-like substance, and it's where all the chemical reactions occur. We have the nucleus here. So I mentioned that earlier. Nucleus is where all the DNA is housed, floating around in that cytoplasm. It controls all the cell's activities. We've got the mitochondria, which also floats around in the cytoplasm. This is specifically for aerobic respiration. Does anyone know what aerobic respiration does? That's very important. It's one of the biggest topics in biology, respiration and photosynthesis. Control cells activities. Yeah, keep telling me the answers, guys. Releases energy. Good. I'm happy everyone says released energy and didn't say produces energy. If you say produces energy, you won't get a mark because you can't create or destroy energy. So you have to say releases energy or transfers energy as well. Well done, guys. Then we've got the ribosomes. Ribosomes are there for protein synthesis. So that means they make proteins. We'll learn a lot about protein synthesis more in paper 2. So those are all the structures in an animal cell. So plant cell has a few things that animal cells don't. There's 3 things that plant cells have that animal cells do not. Can you guys tell me what they are? So what do plant cells have that animal cells do not? Plant cells have, let's see what you guys are saying. Yes, we've got a cell wall. Cell wall. What else are people saying? Permanent vacuole. Amazing. And finally, chloroplast. Yes, well done, guys. You smashed it. Okay, so those are 3 things that only plant cells have. Cell wall. Think of a water balloon being put inside like a box. The box itself is the cell wall. It provides it with structure. It's made out of a substance called cellulose, which provides it with its structure. Then we've got the permanent vacuole, which is basically something you find like in the middle of a cell, and it contains cell sap, which is basically like a solution of sugars and salts. Then we've got chloroplast, which we're going to see again in more detail when we do photosynthesis. This is where photosynthesis occurs. Inside these chloroplasts, you have these green pigments, which you call chlorophyll. Chlorophyll. Yes, yes, yes. Someone didn't go to school for this. I'm loving that, loving that. You'll learn a lot today. So that's probably a sacrifice worth it. Now, if you look at a bacteria cell, remember bacteria cells are prokaryotic. No, they don't have a nucleus. Instead of their nucleus, they have all their DNA just floating around in the cytoplasm. So singular strand of DNA here. And they've got these little rings of DNA which are floating around. These rings of DNA are called plasmids. And they did the same thing as well. They hold all of the genetic material in bacteria. Prokaryotic cells, bacteria have cell membranes. All cells have cell membranes. Cytoplasm, just as usual. Ribosomes, which do the same thing. They produce proteins as well. And they have a cell wall slightly different to the plant cell wall. But if you get given a diagram, those are the structures you need to be able to label. You might have heard of a flagella, flagellum, which is the tail. For AQA, you don't need to know about the flagellum. It's not in the spec. If you check the spec points, you'll see that they don't mention the flagellum. See here? But I think OCI and EdXL do need to know about that. Okay, cool. Let's move on. So those are the different types of cells. So cells, the ones that we've been talking about, these are cells without any jobs. What are the names? What's the name given to a cell which doesn't have a job? Can anyone tell me? Something cell, something cell. Something cell. Unspecialized stem cells. Yes, stem cells. That's what I was looking for, stem cells. So we have stem cells. When you're first made, like from a zygote, when you're first formed, you're made up of stem cells, which don't have a function. Now think about you right now. You have loads of different types of cells. Not all of your cells look the same. Otherwise, you're just going to look like this big blob. You have cells for your muscles. You have cells for your brains. For each of your organs have different types of cells. But we all started off as these a bunch of stem cells. So these stem cells, at some point, must have changed into these different cells that we have, which we call specialized cells. So for a zygote, for a fertilized egg to turn into a human, all of those stem cells need to have converted into specialized cells. And that's what makes a human. Now, this process where they specialize is called differentiation. Differentiation. So differentiation, the definition of differentiation is the process where an unspecialized cell or a stem cell changes to a specialized cells. How does it do that? It basically changes the amount of subcellular structures. What does that mean? Subcellular structures are the things that we talked about earlier. They're like mitochondria, ribosomes. It changes the number of those. Some cells might need to have a lot of mitochondria, for example, for a lot of energy. So each of these specialized cells have these unique things about them. And we have so many, but there's only 6 that you guys need to know about. 3 animal specialized cells and 3 plant specialized cells. So first one is a sperm cell. This is the one that probably gets asked the most because there's quite a few adaptations here. We need to know why This specialized cell is specialized for its function. What do sperm cells do? They're specialized for reproduction. They move the male DNA and connect it to the female DNA during fertilization to produce a new human. How is it specialized for that? This is the type of questions that you'll get in an exam. First thing, it has a streamlined head. You can see here the head is a bit pointy. It needs to swim from the male to the female. Making it streamlined means it can use less energy. It's more efficient. The movement is more efficient. It has a lot of mitochondria. And as you guys said earlier, the mitochondria, remember, that's what releases energy for the cells. So for a cell that needs to move a very long distance, it needs a lot of energy. So it has all of these mitochondria packed, loads of mitochondria packed. It has a tail, obviously, to help it swim. And it's got these enzymes in them. And the enzymes are there to basically digest through the egg cell. So the egg cell has a membrane around it. The sperm cell reaches the egg cell, and the enzymes digests the outside of the egg cell so that the DNA can meet up with the female DNA to produce a full set of DNA. Yes, I'm seeing all the right answers here, guys. Well done on the chat. I'm seeing it all. Next, we've got nerve cells. Nerve cells. We've got nerves all around our body. We learn a lot about nerves and neurons, otherwise known as neurons. We learn a lot about that in paper 2. But they basically send messages around the body. For now, for paper 1, we can just say they send messages around the body. The way they send the messages is using electricity, electrical signals. And imagine cells that need to go all around your body. They need to be very long. They need to have a long shape because they're essentially like wires. You have these wires that go all around your body, which are transporting messages from one part of your body to another. So that's why you can see here, these have a very long shape and they have these connections. They look like branches of a tree, so that one of these nerve cells can attach to another. So imagine this with another nerve cell attached to it and another nerve cell attached to it. And imagine loads of nerve cells attached end to end. It's going to basically form a wire. That's what a nerve is. They've got this myelin sheath. This is just there for insulation. It insulates this long strand, which we call an axon, so that the electric signals can pass through it very efficiently. Yep. Is a myelin sheath made of cells? Yes, it is. It protects the axon. Yes, and it insulates it, more importantly. Yes, smashing it, guys. That's nerve cells. Muscle cells. Not much to say about muscle cells. I'm hoping it's obvious what muscle cells need to do. Muscle cells contract, right? We use our muscles for movement. That means we need it. We need it to release a lot of energy. We need muscle cells to release a lot of energy, right? Exactly. Contract and relax. We need many mitochondria. That's why the only thing I've labeled here is mitochondria. It is packed with mitochondria so that we have all that energy to contract and to move. So those are the 3 animal cells that you need to know about. And there's also 3 plant cells that you also need to know about. First one being root hair cells. We're going to see root hair cells quite a few times today. So I'm not going to go through it in a lot of details now. It's used to absorb water and minerals. It's specialized because it has these hairs that stick out, and that increases its surface area. We will come across root hair cells again. Then we've got the phloem cells. Again, we're going to go through the phloem cells again in the second or third session. Phloem cells, they're used to transport food substances. So photosynthesis makes food for plants, and the phloem cells transports it from one area of the plant to another. And we've got xylem cells as well, which make up the xylem tube. This is a transport system, and it transports water and mineral up the plant. And yes, it's got lignin. So it's got this substance called lignin, which strengthens it because it's made up of dead cells. But I'm going to go through these again in a bit. when we go through this topic. So this is all to do with plant organization. So that's the second topic that we're going to go through. All right. So I hope all of that makes sense, guys. Let's move on to the next section. So remember, we have these spec points. These are the spec points from the exam boards that you can use as a checklist. So as you go through this, guys, just tick the ones you know, and the ones that you need to revise, put a question mark or something, and you can come back to it and revise. You don't need to do that now. You can spend some time after doing it when you're actually doing some revision as well on your own. Right. First, required practical. Required practicals are so important for your exams, guys, because they are guaranteed to come up. That doesn't mean microscopes are guaranteed to come up. Required practicals are guaranteed to come up. In paper one, there's a few required practicals. I think it's about four. Out of them, some of them are guaranteed to come up, and they're guaranteed to make up about, I think it's 30% or 20 or 30% of your marks have to be on required practical. So they're very, very, very important. But that doesn't mean this required practical will definitely come up. All right. So this is all to do with microscopes. Two types of microscopes you guys need to know about. We've got light microscopes, electron microscopes. Okay. And we can see that here. The main difference between the two is that you can basically think of it as electron microscopes are so much more powerful than light microscopes. But there's some key terms that you need to use when you're describing them in an exam. So these are the ones that come up the most: resolution and magnification. Okay, so light microscopes, these are the ones that you use in school. So if you've ever used a light microscope in school, if you've used a microscope in school, it's definitely a light microscope. Why do you think electron microscopes have not been used in schools? What do you guys think? So light microscopes have a lower resolution and lower magnification as well. Exactly. They're too expensive. Electron microscopes are too expensive, and they're complex as well. They can only be used by scientists. They're very complicated to use. Even your teacher wouldn't be able to use. I wouldn't be able to use an electron microscope. So they can only be used by scientists. But they have a very high resolution and a higher magnification. So if you were to look at like a cell under an electron microscope and compare that to the same cell in using a light microscope, this is what you see. If you used an light microscope, all you'd see is the cell and the nucleus. So if you see a diagram with just a cell and a nucleus, that means it's been viewed at using a light microscope. If you were to look at it using an electron microscope, you would see the nucleus, but then you'll see all the other stuff in it. You'd see the mitochondria, you'd see ribosomes, and anything else. So if it's a plant cell, you'd see the chloroplast as well. It gives so much more detail because there's more resolution. There's a higher resolution. Electron microscopes kill the cell. Yeah, but I don't think you really need to mention that in an exam. It's mostly, if you look at the mark schemes for the past papers, it's mostly just talking about cost, resolution, and magnification. So let me highlight those three. Those are the ones that I usually the ones that I. So you could get asked questions comparing the differences between the two. You could also get asked a calculation question here. So this is quite common. These calculation questions are quite common. If you go on our website, you'll see there's quite a lot of magnification questions on there because they've come up quite a lot in the past. In biology, remember, you don't get a formula sheet. You need to memorize all your equations. For magnification, if you can't remember it, use I am. So put an I at the top. I'm here. Because the equation is magnification is equal to image size over actual size. And you can rearrange it. Actual size is equal to image size over magnification, or image size is equal to actual size times magnification. I am. this is how it's given in the spec. So I've written it like this. Size of image is image size. Size of real object is actual size. So if you were given the size of something viewed within a microscope, that would be the image size. The actual size is the size that it is in real life. In these calculations, there's a very important conversion that you guys need to know about, and that's the conversion between micrometers and millimeters. Micrometers and millimeters, okay? If you want to convert from millimeters to micrometers, you times the number by 1,000. And if you want to convert from micrometers to millimeters, you divide by 1,000. It's important that you make sure that every time you use this equation, you have the same units. So you see this question here. It says a plant cell has a length of 30 micrometers. When drawn on a diagram, it has a length of 150 millimeters. We need to convert either this into millimeters or the millimeters into micrometers so that they're both the same. So let's just do that here so I can show you. So it was, let me take a look at it. It was 150 millimeters and 30 micrometers. So if we want to turn millimeters into micrometers for this calculation, what we would do is times by 1,000. So we would do 150 times by 1,000. That's going to give you 150,000 micrometers. Okay, now we can put it into our equation. The equation was asking us to find out the magnification. So magnification is equal to image size over actual size. Remember, I've just used that triangle up there. We're going to put the image size at the top. Let's just note down what the image size was. The image size was the 150. And this was, okay, so we're going to do 150,000 over 30. 150,000 over 30. Okay, and that's what we do in this calculation here. 150,000 over 30, which gives you 5,000. So the magnification of this microscope is times 5,000. That's how much it zooms in to any image that you're looking. Yep, well done, guys. So that's a common calculation, and that's probably the most common type of question you get on microscopes. The other ones you can get are on steps on how to prepare a slide and how to use a microscope. What's the resolution? Someone asks. Resolution, good question. So magnification is how much bigger the image size is to the actual size, or how much you can zoom in. Resolution is how clear the image is. So it's like TVs. If you have a 4K TV, the resolution is much higher. You can see the image much more clearly compared to a HD 1080p TV. And yeah, you can pause and go back, guys, if you do miss something. But I will try to keep the pace. I just want to teach you guys slow enough, but at the same time, finish on time as well. So that you guys aren't here for too long. Preparation of slides. What do you need to do? So you start off with a slide. You add a drop of water onto the slide. And usually they ask you about onion cells, how to observe an onion cell. So you take out a thin layer of onion cell. We call that the epidermal tissue. So cut up an onion, use tweezers to peel off a very thin layer of the epidermal tissue on the onion and put it on your slide, which has a drop of water on it. So water here, put the epidermal tissue on that. So that's what you're trying to view, those onion cells. Now, most of these cells are completely see-through. They're colorless. So if you look at them through a microscope, you won't see anything. So what we need to do is stain it. So to stain it, we use iodine solution to stain it. And then you place a cover slip on top, which is basically just another piece of glass, another sheet of glass. And you have to make sure there's no air bubbles in it, because that will affect how clearly you see your image. Okay. No, it's not on the spec. The thing with required practicals, if you look at the spec points and required practicals, they're usually just like 2 lines, and they're not, then they don't have a lot of information at all. But it is usually onion cells they ask you about. That's a good observation that you made there. How do you use a light microscope? Always start with the lowest objective lens first. The lowest powered objective lens. Okay, so look at this. This is a diagram of the microscope. We've got two different lenses here. We've got the eyepiece lens at the top. The eyepiece lens only has one magnification, whereas the objective lens has three magnifications that you can switch through. So the top eyepiece lens, it might be, I don't know, let's say ×5 magnification. Yeah. And the objective lenses, they'll have three different ones. They'll have a low powered one, so maybe like a ×5, and then a ×10, and then a ×20 magnification. And what you can do is you can switch between these three magnifications based on how much you want to zoom into the sample. So for example, if you used a ×5 objective lens, the magnification you've got is actually ×5 times another 5. You need to multiply the numbers by each other. So if you were to use the ×5 magnification objective lens, it would actually be 5 times 5. You need to multiply it by your eyepiece lens magnification. So that'll be ×25. If you use the ×10 objective lens, that's going to be ×50, because 10 times 5 is 50. And if you use the 20, that's going to give you a ×100 magnification. Okay, so just remember that. Okay, someone trying to ask a question? Let me know what it is. I am trying to keep up with these questions. Forceps instead of tweezers? Yes, that's fine as well. What's the wheel? The wheel, I'll mention that later. I'll talk about the wheel later. Yep. Okay, cool. Then we've got the stage. Yes, the stage is where we put the sample on, and underneath that we've got light, which shines onto the stage so that you can clearly see it. You've got the adjustment knobs here, which you can use to zoom in and out. This will bring the stage up and down. So if you turn these adjustment knobs, it's going to move the stage up and down. That's just going to help you focus. The adjustment knobs are there to help you to focus. So start off with the lowest powered objective lens, and then you use your adjustment knobs to help yourself to focus whilst looking in the eyepiece lens. If you want to zoom in more, you then switch to a high objective lens. So you always start off with the smallest one. So if you want to really look into a cell, you always start off with the ×5, and then you switch to ×10, then you switch to the ×20. You don't go straight into the ×20, because the ×20 lens will be a really big one, and it could smash when it hits the stage. I've seen that come up in an exam question before. Some microscopes have lights. Yep, some microscopes have lights instead of mirrors. That's true. Stage clips hold slide in place. Yes, they do. The mirror can act as a light. Yes. So these light microscopes can have a mirror or a light. Would you write all of that for a 6 marker? Yes, pretty much. Pretty much. All of that would get you 6 marks there. What were you asking, Frozen? Stage clips. Yeah, stage clips. Is that what you're asking? When you're moving the stage, you look from the side. At the start, you look from the side, but then you need to focus on it. To focus, you need to look through the eyepiece and then turn the knobs so that you can see it. I hope I'm answering all your questions, guys. And then you could be asked to look at a drawing as well, or talk about a drawing. I doubt they'll ask you to do a drawing yourself, but when you're making a drawing of a cell based on your microscope image, make sure you use a pencil. Use unbroken lines. So basically, don't do what you do when you sketch. Don't do this. Yeah, some people do that when they sketch. I've never seen a question where they ask you to sketch. So I don't expect them to do that, because there's so much room for like error there. But you might be asked to mention it, like they might give you a student's drawing and ask you to say what they've done wrong. So they have to use pencil, use unbroken lines, and then try and draw proportionally. But I doubt this will come up. Okay. Exactly. It's not an art exam, yes, but you have to be as accurate as possible. Okay, what else are people asking? What is a magnetic level? A magnetic level? Magnetic? And the magnetic level. I think that's a typo. Oh, not magnetic level, magnification level. That's a good spot. Magnification level. So how much it's been magnified. So times 1,000, times 100, something like that. Good spot. Do we have to identify the errors in someone else's drawing? Again, I haven't seen that, but I'm guessing that's how they could test you on it. That is what they could ask. They could ask. Why do you add water to the slide? Is it unnecessary? When I see caps, I just imagine the person just shouting at me. It is because that's the only way the second slide, the cover slip, will stick on. It needs water to stick on, and then it's not gonna flatten the cell. So you should, but you might get away with that without saying adding the water at the start. Will the objective lens always match the magnification of the eyepiece and lens? No, they won't match. They could be different. You just need to multiply the two numbers together, Jathona. Do we need to hazards? Not really for microscopes. Oh, sorry if you asked 20 times. My bad. I didn't see it. I am trying to keep up with the questions, guys, so bear with me. What feature gives it a greater field of view? Zooming out gives it a greater field of view. All right, cool. Let's move on. This is a triple only topic, so we'll do this at the end. Culturing microorganisms, a very quick one anyway. And we'll move on to chromosomes and mitosis. How are we doing for time? We're about, wow, time's flying. 35 minutes. Let's see. Chromosomes. What are chromosomes? They contain DNA. All right. We are going to cover chromosomes in paper 2 as well. They're DNA which has been coiled up. DNA is basically long strands of genetic code, and chromosomes is that coiled up into these structures. So human body cells have 46 chromosomes, memorize that, and they exist as 23 pairs. So here we've got 46 and 23 pairs. Half of the pairs come from the mother, half of the pairs come from the father, and putting them together, you get 46. So sex cells, you have sperm cells, which we've already talked about. You have egg cells as well. These are known as gametes, and gametes have half the chromosomes. They only have 23 chromosomes. So when the 23 chromosomes from the sperm cell meets up with the 23 chromosomes in the egg cell, it forms a human with 46 chromosomes. That's how it works. And the chromosomes have genes in them. Again, you get asked more about genes in paper 2. But for now, I'm just going to say genes, they are responsible for your characteristics. So they make you who you are. So the way you look, the way you act, the way your body works is all to do with your genes. You have several different genes in your body, which is all within your DNA. Okay. The sperm and egg, are they called haploids? They're not called haploids. The number, the 23 number within them is called the haploid number. They're not called haploids. They're called gametes. Yeah, so Down syndrome is when a person has an extra chromosome. You're right. But you don't need to know that for your GCSEs. Each gene codes for specific amino acids. Yes, so that's more for paper 2. That's more for paper 2. Okay. Diploid is just the number. So 46 is the diploid number and 23 is the haploid number. But again, I think that's more of an EdXL thing, not really a AQA thing. So what you do need to know in more detail is the cell cycle. So cell cycle is basically how a cell reproduces, how you go from one cell to it splitting into 2 cells. And this is quite a simplified approach that you need to know for GCSEs. There's much more to it in real life, but you only need to know it as 3 stages. 1, 2, 3. First one is growth and DNA replication. Second one is mitosis. Third one is cell division. All right. So growth and DNA replication. So before a cell splits into two, it needs to double everything inside of it. Because imagine you have a cell, let's say it had let's say it had 10 mitochondria, and then it split into two, then each of the two cells are only going to have 5 mitochondria. That cell hasn't reproduced. You've just got half of a cell now. You've got 2 halves of a cell. So before it splits, you need to double everything inside of it. And that's what growth and DNA replication is all about. So it doubles everything in the cell, including the DNA. The DNA inside needs to replicate. You've got these single chromosomes, and they replicate to form X-shaped chromosomes. Okay. Is meiosis different? Yes, it is. We'll learn that in paper 2. If you're AQA, you don't need to know about interphase, metaphase, and all of that, by the way. You just need to know these 3 steps. If you're EdXO, you do need to know about it. Spindle fibers, I haven't seen mentioned on the spec. I call them cell fibers in the notes because that's what they've been referred to as. Yeah, exactly. So once everything has been doubled, all the cells all the chromosomes, line up in the middle of the cell. And you can see that there. They're all lined up in the middle of the cell. And these strings, you can call them strings, they're cell fibers or spindle fibers, as some people have said. They pull apart the X-shaped chromosomes. So they pull them apart, so each half goes to either side of the cell. This process is mitosis. Now what you've got is the correct number of chromosomes on each cell. So you start off with 46 chromosomes, double everything, you get 96 chromosomes. Then you split the 96 chromosomes so that you've got 92. When you double 46, you get 92. You have 46 chromosomes here and 46 chromosomes here. So these are ready to be individual cells now. Now, the cell membrane splits into 2 and the cytoplasm splits into 2. And you've got 2 cells which are identical to the first cell you started off with. These are clones of that first cell. We call these daughter cells. They're identical. They have the correct number of chromosomes. They have 46 chromosomes now. And this process just happens over and over again. So from the moment a sperm meets an egg and it turns into a zygote, it basically starts off as a single cell like this. It divides by mitosis. So 1 cell turns into 2, 2 turns into 4, 4 turns into 8, and it keeps dividing while you're in your mother's womb. And then slowly those cells start to differentiate, like I talked to you about earlier. It starts to turn into specialized cells and it forms a human slowly. So this last process, yes, it's called cytokinesis. I see some of you typing that in the chat as well. Does cell membrane and cytoplasm splitting happen stage 2 or stage 3? Cell division is stage 3, also known as cytokinesis, but that's not in the spec. You don't need to say that word. Why is it easier to view cells with lower power objective lens first? It's not that it's easier to, because you get a wider field of view, so you can see everything, and then you zoom in. If you were to go straight to the highest power objective lens, it's going to zoom in somewhere random, and you're not going to know where you are. It's just common practice. And the other thing is the big objective lens could break if you use that straight away and it hits the stage. Do we need to know examples of different cell sizes? No, you don't. No, you don't. So a question. No. Oh, an herbicide can disrupt plant cell division. That's the paper 2 question. Paper 2 question. It's just what herbicides do. They disrupt how plant cells can divide. Is micrometers to mm the only conversion we need to know? No, it's not. It's the most common one. You need to know centimeters. You need to know meters. In biology, you won't need to know, I don't think you'll need to know kilometers. I've never seen kilometers come up. Centimeters, millimeters, meters, and micrometers. Those four, I'd say. Nanometer is 10 to the power of 9, if you want to know. 1 nanometer is 10 to the power of 9 meters. That's more in physics. That's more in physics. Interphase, like I said, you don't need to know interphase. What is the definition of mitosis and cell division? So mitosis is the process where they don't ask what the definition is. They ask what the process is. But yeah, it's a process where a cell divides to form two identical daughter cells during cell division. Okay. How are twins formed? That's another question. That's a paper 2 question. You don't actually even need to know for paper 2. That's not something you need to know for your GCSEs, don't worry. Okay, so we've got the summary here of all the steps that I just talked to you about as well. Any other things I missed out? The 2 identical daughter cells are formed at the end. Subcellular structures are copied, mitochondria, ribosomes. The nucleus replicates to form X-shaped chromosomes. The membrane of the nucleus breaks down. The chromosomes line up at the center and the cell fibers pull them apart. Then new membranes form around each set of chromosomes, so to form the 2 new nuclei. And then the cytoplasm and the cell membrane split to form 2 daughter cells. Okay, stem cells. So I've told you what a stem cell is already. A stem cell is something, it's a cell that's undifferentiated. We need to know the types of stem cells that we can have. So humans have 2 types of stem cells. We have embryonic stem cells, which we get from human embryos. Like when you were first formed, you were just stem cells, right? But we also have adult stem cells. So once you've turned into a child, you basically lose all your embryonic stem cells. You've only got adult stem cells, even though you're technically still a child. They're found in the bone marrow of a human. So that's in the center of your bone. You find bone marrow. The difference between the 2 types of stem cells, human stem cells from embryos, can turn into any type of cell. They can differentiate into any cell. Remember what I said? I said stem cells differentiate into specialized cells. So human embryo cells can turn into any cells, whereas bone marrow cells, the adult stem cells, can only turn into blood cells. It can only differentiate into blood cells. So that's that's something that's not great about humans, but it's the case. At least we can create our own blood cells. So adult stem cells can be used in medicine. We use it in medicine. We can use it to treat diseases like diabetes and spinal injuries. So if someone has a pancreas that isn't working properly and has diabetes because of that, we can use embryonic stem cells to create new pancreatic cells, new pancreas cells, which makes their pancreas work better. And what you can do is use something called therapeutic cloning. Therapeutic cloning is when you make a clone of a person in the form of an embryo. So imagine getting your DNA and creating an embryo, a clone of that. What you essentially have is an embryo with exactly the same DNA as you. So you can extract the embryonic stem cells from that embryo, and then you can use that to treat any diseases that you may have. It's quite good because the person will not have any rejection. The body will not fight against you trying to implant any organs from something that has the same DNA as you. Right, let me see these questions. So are we talking about mitosis still? What are we talking about here, guys? So mitosis, you start off with 46 chromosomes. The daughter cells also have 46 chromosomes. They're identical. The cells you get at the end of mitosis is exactly the same as the one you start off with. Is that answering the question? Is two daughter cells are formed? Yeah, they have 46 chromosomes. Is that what we're asking right now, guys? Yeah, exactly. Genetically identical. They're copies. Imagine copying and pasting the original cell. That's what mitosis does. Okay. So therapeutic cloning, you don't need to know the full process of it. Yeah. All you need to know is it creates embryos with the same DNA as a patient. And it's good because it reduces rejection. The thing with this is that there's ethical considerations. People are against therapeutic cloning because what you're doing is you're creating an embryo and then you're using it for its cells and you're basically destroying it. They get destroyed at the end of it. So people are against it because they think that the embryo is a potential human life and you're wasting it. But some other people are for it because they think that the fact that you can save someone else's life using the embryo from suffering patients, it's worth it, basically. So these are just some ethical questions you might get asked in your exams. That's what you can say there. Why people are against it and why people are for it. Playing God, yeah. It's a bit vague to say playing God. Yep, blue G8, that's right. You start off with 46, it replicates to form 92, and then it splits to form 46 again. Yeah. Is healing with stem cells the same as therapeutic? Good question. No, you don't need therapeutic cloning. You can use other stem cells to heal it, but therapeutic cloning is a very specific time. Plant stem cells. We got meristem tissue, so that's where they're found. So humans, you can have embryonic stem cells or adult stem cells in the bone marrow. Plant stem cells, they're found in a tissue called the meristem tissue, and you can find those on the tips of plants, shoots and roots. So right on the tips, that's where it's growing. That's where you find meristem tissue. These can differentiate into any type of cell, so not just specific cells. Like we have adult stem cells that can only differentiate into blood cells. Plant cells can differentiate into any cell, and they can differentiate at any point in their life as well. So it's very useful. The reason why we use these is if we want to clone a plant, we can. So if there's like a rare species of plant that's about to die out, we can protect them by producing loads of clones of them. We can also use it to produce disease and insecticide resistant crops as well, the ones that don't die when disease hit them or when we use pesticide on them. Okay. Yes, meristem tissues are areas of rapid growth. Yeah. Okay, last thing, guys, is diffusion, osmosis, and active transport. And what I'm going to do is I'm going to teach you all three in one go, because if you know one, I'm telling you, it's easy to learn all of them. Okay, they're pretty much very similar. Okay, so diffusion is when particles move from one area of high concentration to an area of low concentration. Okay, so if you spray perfume in one corner of a room, those perfume particles will spread out to the rest of the room. That's the definition of diffusion. For osmosis, it's basically the same. So look at what I'm going to do. I'm going to literally copy and paste this. And I'm going to change it because I'm going to change it slightly, because you can think of osmosis as diffusion with water. So I'm going to say the movement of water particles. I'm just going to add the word water here. Water particles from an area of high water concentration to an area of low water concentration. And one extra bit that we need to say here We've got the M in osmosis. That helps you remember. There's a membrane in there. So through a partially permeable membrane. So if you have diffusion occurring with water, it's known as osmosis. It moves through a partially permeable membrane, which basically is a membrane, so like a skin or something with small, tiny holes in it that only water particles can pass through it. Okay, so it's kind of like a sieve. A sieve, you can only let water pass through it, or the large particles get stuck in the sieve, right? So osmosis has a partially permeable membrane, which only allows water to go through it, but other particles, larger particles like proteins or sugars, can't pass through it. Yep, I'm going to go through the required practical in a second. Active transport. Active transport, you can think of as the opposite of diffusion. I'm just going through these definitions first. So we're going to copy and paste this again, and I'm going to change one thing. What do you guys think we're going to change here for the definition of active transport? Movement of particles from an area of, if it's the opposite of diffusion, all I'm going to do is I'm going to swap this round. Exactly. If you know one of these, it's so easy to remember the others. So area of low concentration to an area of high concentration. And what you should also mention for this is that it uses energy. Using energy from, where do we get energy from? We said it earlier, respiration. So all three of these, you learn it as three different topics in school, but if you know one, the others are very similar. So diffusion, movement of particles from an area of high concentration to an area of low concentration. Osmosis is diffusion with water. It's the movement of water particles from an area of high water concentration to an area of low water concentration through a partially permeable membrane. Active transport is the movement of particles from an area of low concentration to a high concentration, and it uses energy from respiration. Okay, here some of you might have seen, or you might have been taught it as from a dilute solution to a more concentrated solution. That is fine as well. They mean the same thing, concentrated solution. Okay, so those are the definitions. So for diffusion, you need to know the factors that affect diffusion. The factors that affect it is the concentration gradient. So diffusion goes down a concentration gradient. The bigger the difference in concentration, the faster that diffusion will happen. So if you have a very high concentration and a very low concentration, diffusion will happen faster. If the temperature is higher, again, diffusion will happen faster. And if the surface area of the membrane you're talking about is bigger, it will happen faster as well. You could say high water potential. Yes, you can say high water potential as well. Is diffusion and osmosis passive? Yes, good question. Diffusion and osmosis do not use energy. They're passive processes. Yes. Is dilute to a low concentration? No, it's dilute to a concentrated solution. Concentrated, more concentrated solution. Because dilute basically means it has a high water potential or high water concentration. Surface area to volume ratio is something that you'll see quite a lot here, okay? The bigger an organism is, the lower its surface area to volume ratio. So you can see here, this elephant has a low surface area to volume ratio. This mouse has a high surface area to volume ratio. And you could get asked to prove this. And you can do that. This is basically maths, by the way. I've seen this question before as well. It's come up a few times where you need to work out the surface area to volume ratio of cubes. So this cube here, to find out the volume, you do base times height times width. So 1 times 1 times 1 gives you 1 meter squared. 1 meters cubed, sorry. Let me put a cube there. Surface area, you do the area of one face. So that's 1 times 1. the area of the square, and multiply it by the number of faces there are. In a cube, there's 6 faces, so that's going to be 6 meters squared. So the surface area to volume ratio is 6 to 1. You can do exactly the same here with 2, and you get 8 meters cubed and 24 meters squared. That will give you a ratio of 24 to 8. Now, if you divide both of these numbers by 8, you can simplify it to a 3 to 1 ratio. So this proves that the bigger the cube is, the smaller the surface area to volume ratio. So this is basically the elephant, and the other one is the mouse. And we've just proved that the elephant has a smaller surface area to volume ratio. So there are a few exchange surfaces that you guys need to know about. You can't understand surface area to volume ratio. It's a calculation. It's literally just a calculation, okay? If you were to work out the surface area of a cube and then the volume of the cube and divide and then write a ratio of them, that is surface area to volume ratio. It's just maths, yeah. So what time does it finish? It's meant to finish at 11. I still have a little bit more to do. Let's think what I can do. We have this to go through. Oh, we're nearly done. We're nearly done. We're nearly done. No, we're not. We've got more. Okay. What we'll do, we'll take the break now. We'll take our first break now, guys. And we'll carry on in the second session with these exchange surfaces. All right. Any other questions, guys? So we'll be back at 11:15 for the second session. Let me try and answer these questions before you guys go on your break as well. Will they always give a very regular shape? They'll always give you a cube. They'll always give you a cube. What does it mean, concentration gradient? Concentration gradient is the difference in the concentration. So if you have a concentration of 10 on one side and a concentration of 2 on the other, the concentration gradient is 8. So the break, yeah. We'll have a break now for 15 minutes, guys. 15 minutes. I won't go through triple topics now. No, I'll save that for the next break, okay? Because organized topics. So triple, guys, you can have your break as well. Everyone can have a break now, and we'll save it for the triple stuff for the next one, okay? So have a good rest, guys. Go stretch your legs and be back in about 15 minutes. Can we remember it as ice cubes as the one that's smaller melts first? Yeah, Jatana, that's good. That is a good analogy. That is that's a good way to think about it as well. Yeah. Okay, good to hear, guys. Good to hear that this is useful. We'll carry on, get some rest, and we'll carry on in about 15 minutes. Microbiology practical, I'm not sure what that is. That's not in AQA, so probably won't go through it. Yep. Okay, good, guys. That's good to hear. I'll see you all in 15 minutes. Go and get some rest. Welcome back, guys. Can you hear me okay? How are we finding the session? I'm trying to find the balance between getting everything done and also trying to make you guys understand everything as well. I'm trying to find the balance, guys. Is it all good? Amazing. That's good to hear, guys. Like I said, it's all free. We're trying to make this as useful as possible for you guys. So good to hear that it's useful. So yeah, guys, some of you asking about the cram classes. So let me put the link in for the cram classes. So these are sessions that we're going to do the day before your exams. They've been quite popular in the past. So literally the evening before the exams, we do a revision session, and all the paper ones are free again. I'm going to put the link in the chat right now, and you guys can sign up right now. You don't lose anything by just signing up. Sign up to the free ones. If you're in FRL classes already, you don't need to sign up. You're going to get those free if you've got the subscription. But you guys might as well sign up now. If you've already signed up, then that's fine. You don't need to do anything. And I'm going to spin a wheel again at the end of today as well. And someone else is going to get all of that for free, including the paper twos for this Easter Bootcamp and all the cram classes as well. So if you haven't signed up, then you can put your name down on that using that link there. All right. So let's carry on. Yeah, Minto play one. That was Dylan, if I remember correctly. It was Dylan, right? Congratulations to you. Okay, guys, we've got a lot to get through. We're still on topic one. So let's carry on. Where I left it last time, I said the bigger an organism is, the smaller its surface area to volume ratio. So small organisms like bacteria, they have a very large surface area to volume ratio. That means their diffusion is very good. They can diffuse substances within them very easily. They can just go up to a substance and it'll go into them. We can't do that because we're large organisms. We need to get substances from the outside using different methods. We use something called exchange surfaces. So how do we get oxygen within us? We use our lungs, right? Bacteria don't do that because they're so small. They can diffuse oxygen directly into their bodies. So there's 4 exchange surfaces that we need to know about. So we've got alveoli in the lungs, leaves in plants. We have gills in fish, and also we've got the small intestine, which contain these little cells called villi. The questions that you get asked on this are to do with these things. Why are they adapted for performing their function? Why are alveoli good at, for example, taking in oxygen? Why they're adapted? And these are the things that we need to talk about. These 4 things here: surface area, the fact that they have a thin membrane, so they have thin walls to make diffusion easy. They have a good blood supply. Some of these have a good blood supply to help you get substances into the blood quicker, and whether they're ventilated. Ventilated just means they have a good supply of gas. So alveoli, for example, have all 4 of these. They have a large surface area. That's why they're not round. They have this cloud-shaped surface. They have very thin membranes to make diffusion quicker. They have loads of capillaries around them to help the oxygen diffuse into the blood easily, and they're ventilated. They have a lot of oxygen being pumped into them while we're breathing. Yeah, you can say 1 cell thick or surrounded by capillaries. Exactly. Yeah. Leaves are very thin. They're flat. They have a large surface area. They have thin membranes as well. You don't really talk about blood supply for leaves because they don't have any blood. Plants don't have blood, and ventilation as well. You could talk about ventilation, but it's really the air. It's not well ventilated. They're in contact with the general atmosphere. Villi have a large surface area, so these are in the small intestine. They're used to absorb nutrients into the blood, so they have a good blood supply. They also have a wall that's one cell thick, so they have a thin membrane. They don't need to be ventilated because there's no gas exchange here. It's just exchange of nutrients. Then we've got the gills in fish, which have a large surface area. They contain these structures called lamellae, which we have here. They have thin membranes again. They have a good blood supply. A lot of blood flows to the gills of a fish, and they're ventilated. A lot of oxygen can come in contact with it. Oxygen in the water. Yeah. Ventilated just means they have a good supply of oxygen for these two examples, for the alveoli and gill. I hope that answers your question. So diffusion, that's everything you need to know in diffusion. Osmosis, I told you the definition of osmosis. Osmosis is basically with water. It's diffusion with water. So we can see here a diagram here. We've got a root hair cell. Remember we talked about that earlier. Root hair cells are used to absorb water and mineral ions. So to absorb water, they use osmosis, because remember, osmosis is diffusion with water. And they go from a high water concentration in the soil to a low water concentration in the cell. They go down the concentration gradient. And they go through a partially permeable membrane here in the root hair cells. That's how these root hair cells take up water. It's a process of osmosis. So that's one of the things root hair cells do. They take in water. For water, they use osmosis, but they also take in something else, and they don't use osmosis. They use mineral ions. They take in mineral ions. What did they use to take in mineral ions, guys? So for water, they use osmosis, and for mineral ions, they take in mineral ions using active transport. Well done. So the reason why they need to use active transport is because in the soil, let me show you a diagram. I'm going to come back to that second. In the soil, you can see they have a low mineral ion concentration. In the soil, you have a lot of water, but you don't have much ions. You don't have many minerals in the soil. So it needs to go from a low concentration to a high concentration. You can see here they've got loads of mineral ions in there. It needs to go against the concentration gradient. It needs energy for that, so it uses active transport for that. Low to high, exactly. You need to know lamellae. Everyone needs to know lamellae. Yes, everyone needs to know lamellae. Okay, required practical number 2, osmosis. This is the one where you use a plant tissue, such as potato or some sort of carrot I've seen in the past, and you put it in a solution. And what happens is if you put it in a solution with a high water concentration, the mass will increase. That's because the high water concentration makes the water go into the low concentration of the potato. The potato cells have a very low water concentration. So the water goes in by osmosis from the high water concentration to the low water concentration, and it makes the potato get bigger. So its mass increases. If it's the other way around, if you have a very concentrated solution or a low water concentration, then the water leaves the potato cells, and that causes it to shrink. So you need to understand that concept. Now, what can they ask you? A very common calculation is a percentage change calculation. So let's say you do this experiment. You have an initial mass of 1.3 grams of the potato, and you put it in a solution for 24 hours, and then after that, it becomes 1.51 grams. So it's increased in mass. And they might ask you to find the percentage change. And this is not just in this topic. They could ask you percentage change calculations in physics, in chemistry, even in maths. It's a common thing to get asked. So what you do here is you do the change in mass over the initial mass. So the change in mass is the change between these two numbers. So we had 1.51 and 1.3. So we're going to do 1.51 minus 1.3 over the initial. What did we start off with? We started off with 1.3 times that by 100. If you do that, you will get the percentage change. And if You put that in your calculator, you should get 16.2% when you times it by 100, 16.2%. Yeah, exactly. Some things that you need to mention in this practical is make sure that the potato cylinders have similar size. They come from the same potato. And you know, when you weigh them, when you take them out the solution, make sure you blot them dry with paper towels. You want to remove all the excess water because it's going to make your measurements less accurate. So you need to make sure as soon as you've taken them out of the solution, you blot them dry before weighing their masses. All right. So that is that practical there. They could ask you for the setup. They could ask you to describe the method. They could ask you for a calculation. They might ask you to draw a graph as well. If you have a scenario where the mass doesn't change, it means the solution. So let's say we put this in at 1.3 grams, and at the end it's still 1.3 grams. So the mass hasn't changed. That tells you that water hasn't gone in or out. Is net. The net movement of water is 0, which means the concentration, the water concentration outside is the same as the water concentration of the potato itself. Yeah, you can use eggs as well. Yes, you can use eggs as well. What would you use to cut the potato? You can just say a knife. Can you say measure in the same size whilst cutting it into cubes? You can do cubes as well. Usually cylindrical. They at the start they have to be similar. If you're measuring the percentage mass change, it doesn't matter if they're slightly different masses, but you need to try and keep them the same as possible, as similar as possible. You don't need to know those words, Alexi. No, you don't. Yes, the projections, the lamellae are the projections, Dennis. Control variables, things like temperature of the room, you need to keep that constant as well. Has the mass been lost or gained by osmosis? It depends on the concentration outside the water. If the water concentration is greater outside, then it's going to be gained. And if it's less, it's going to decrease in mass. Summarize the steps. Here we go. Cut the potato cylinders into similar size. Immerse them into different sugar solutions. Yep, make sure they have different concentrations. You're going to have one with pure water because you're going to use that as a control so that you can compare it. Before you put it in, you have to measure the mass using a mass balance and then wait a couple of hours, maybe 24 hours. That's what I put in the notes. And then you blot them dry with a paper towel and then weigh the mass again. Okay. All right. And then active transport, I've explained active transport and I've explained active transport in root hair cells. There's just one more you need to know for active transport, which is the fact that we use active transport to absorb glucose in the small intestine, because in the small intestine, in the gut, there's a low concentration of glucose, but in the blood, there's a high concentration of glucose. So it has to go against the concentration gradient. So for that, we need active transport. So yes, that is everything for active transport that you need to know. That doesn't come up much, that one. All right. So I think we're done with topic 1 now, guys. So remember, at the end, you get exam questions you can try out. We'll see if we have time to do exam questions, but biology is very theory heavy. So my main aim is just to get through all the theory with you guys. We will send you guys mark schemes to these as well. Okay, so let's go. I don't have a memory trick to answer osmosis practicals. No. Triple content. So the thing with paper 1 biology, there's not much triple content. Okay, there's only culturing microorganisms and monoclonal antibodies and plant defenses. There's only 3, so I can get through that quite quickly. We've got 4 sessions, so I'll do it I'll try and do it just before lunch, some of the triple content. And the last topic in biology is quite short as well, so that's just respiration and photosynthesis. So I can do it there as well. Right, topic 2 is the biggest topic in biology paper 2, principle of organization. So this is about cells, tissue, organs, and organ system. Okay, so the structure goes, memory trick, C2. you have cells, which are the building blocks of living organisms. If you get loads of cells, loads of similar cells working together, you get a tissue formed. And then loads of different tissues working together forms an organ. And loads of different organs working together forms an organ system. That's the order. All right. Many cells, many tissues, many organs form an organ system. Not much to say about that. So this topic is all about basically organs and organ systems. Exactly, Blue. That's a perfect definition there. So enzymes. What are enzymes? Enzymes are biological catalysts. Catalysts are things that speed up the rate of a reaction, and they don't get changed in the reaction, so they're not consumed or altered. Now, they're proteins, which means they're made up of amino acids. And what they do is they fold up into these unique shapes. They have these really weird shapes. You can see here, this is a very simplified diagram of an enzyme. And they have these weird shapes with a structure called an active site in it. And the active site is where the reaction happens. So they speed up reactions. So the reactants of the reaction are called substrates. They basically fit into the active site. And you can see here, it perfectly fits in. We call this the lock and key mechanism. This is the lock, and this is the key that fits into the lock. And only one key can fit into a lock. That's why we call it the lock and key mechanism. It's specific. So each substrate is specific to an enzyme. So what happens is the substrate binds to the active site. It goes into the active site, and then it breaks up into the products. So you can see here, it's turned into a yellow and a green particle. After that, this is important because after that, these two products leave and the enzyme hasn't been changed or altered. So that means a new substrate can now come in and do the same thing. So these enzymes can keep speeding up reactions over and over again because they don't get used up. All right. So to make these reactions faster, there's two factors that affect this whole process. Temperature is one of them. pH is the other. So if a reaction is happening in really cold conditions, the particles aren't moving around very fast. But if you increase their temperature, what's going to happen is these particles are going to be moving around much faster. They're more likely to have the scenario where the substrate goes into the active site. So that's why in a graph for temperature, if you have zero temperature, there's no reaction. The rate is very slow. But as you increase temperature, there's more and more collisions between the substrates and the active site, which means the reaction is going to be faster. So that's why the rate increases and increases and increases. So loads of people think it just keeps increasing and that's it. So if you have like 1,000,000 degrees, the rate is going to go really fast. But that's not real. What actually happens is there's this optimum temperature. Can anyone tell me what they think the optimum temperature is in the human body? That's where the temperature is the fastest. Sorry, the rate is the fastest. So enzymes are working their fastest at the optimum temperature. In the human body, thank you guys, it's 37 degrees. 37 degrees C or 37.5 degrees. That's where they work the best. And you can see past the optimum temperature. So if we were to label this diagram, this would be 37. Past 37 degrees, the rate goes down. The reason for that is because the enzymes denature. Basically, they change shape. So look at that. The active site has just changed shape. And now this substrate is no longer fitting inside of it. So that reaction is not going to be sped up. It's not going to be catalyzed. So the rate goes way down, 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 because the enzymes denature and the substrate can no longer fit into the active site. That's why if your temperature goes up to like 42, you feel really ill. We need to maintain our temperature at 37 degrees for these reactions to happen the most effectively. Can the enzyme denature if the temperature is too low? No. People are saying yes, no, it does not denature. The reason why it's slower is because there's fewer collisions. If the temperature is lower, they don't denature, they just move around slower. So the reaction's slower. They only denature if the temperature is too high. Yeah, exactly. Don't get that confused. For pH, for example, on the other hand, it can denature. So for pH, there is an optimum pH, usually pH 7 around the human body. That's where reactions happen the fastest. If the pH is too acidic, the enzymes denature, and if it's too alkali, the enzymes denature as well. Almost all our enzymes in our body have a pH of 7, apart from, I'd say, there's one that you need to know that doesn't have a pH of 7, sorry, and that is in our stomach. We have enzymes in our stomach called pepsin. And if you guys know about our stomach has hydrochloric acid in it. So we basically have a graph in our stomach which looks more like this, where the optimum is around 2 or 3. 7 is here and 14 is up here. So in our stomach, we have hydrochloric acid because we have this enzyme called pepsin in it, and it works best. Its optimum pH is around 2. And that's when the rate of pepsin is the fastest. The rate of pepsin reactions are the fastest. Everywhere else in our body, for GCSE students, you just need to know it as pH 7 as the optimum pH. All right? Bio is not an enzyme, no. Enzymes do not denature at low temps. Exactly. Right. People are talking about bio and digestive systems. So let's go through that. Digestive system. You need to know the 3 digestive enzymes that you can have. They are carbohydrates. An example of this, you might have heard of it as amylase. Yes, amylase is a type of carbohydrates. We have protease and we have lipase. The one I just told you, pepsin, is an example of a protease enzyme. Lipase, you don't need to know any examples of lipase, but each of these break down a certain type of food. So carbohydrates enzymes breaks down carbohydrates. It's in the name. It breaks it down into simple sugars. Protease breaks down proteins into amino acids. Lipase breaks down lipids into 2 things, glycerol and fatty acids. And you can find these at different positions in the digestive system. So, memory trick here for the digestive system, muscle pier, as in muscle, and pier, you know, the thing that you find in a beach on the near the sea, is not my greatest memory trick, but some students find it useful. So the food starts off in the mouth, where you chew your food. You've got amylase, which breaks down carbohydrates such as starch into sugars. Then you swallow your food, and it goes down the esophagus, which is basically a tube. You don't need to say much about the esophagus. It's just a tube that links the mouth to the stomach. The stomach, we do need to know a bit about. So, like I said earlier, it produces hydrochloric acid. The reason it produces hydrochloric acid, guys, is not because it breaks down food. Don't say that. It produces hydrochloric acid because it provides the optimum pH for pepsin. It makes pepsin work its best. That's one thing. And the other thing is that it kills bacteria. The acid kills bacteria. It also churns the food. It's got muscular walls that break the food down. They contract and relax and break the food down. Peristalsis you don't need to know about for AQA, but that's in the esophagus. Yes, exactly. Next, we've got the liver. Well done. Liver produces something called bile. Bile is not an enzyme, guys. Bile is a chemical which breaks down fats, but it doesn't break it down into fatty acids and glycerol. It just breaks it down into smaller, tiny droplets. It increases its surface area so that it can be broken down even more easily. This bile neutralizes acids that come out of the stomach. Remember I said the stomach has acid. Imagine all that food coming out of your stomach. You need to neutralize it. You don't want the acid around your body. So the bile comes in, which is an alkali, and neutralizes it. which makes it a pH 7 for the rest of the digestive system. So two things that the bile does. First one is it emulsifies fats. It breaks down fats into smaller droplets. And the other thing is it neutralizes stomach acids. Yes, yes, yes. Everyone's got it right there. Then we've got P, which is for the pancreas. Pancreas, very simple. It produces all three enzymes, the carbohydrates, the protease, and the lipase. Then we've got the two intestine. We start off with the small intestine. This is where the villi are. I talked about that earlier. The villi is where food is absorbed. And also it produces the three enzymes. So it produces all three enzymes, and it's where absorption happens. Then we've got the second intestine, the large intestine, where water is absorbed. And then you've got the rectum, where everything that's left over is stored before you go to the toilet, or the feces is stored. Okay. If you're edX, I'm pretty sure you don't need to know the digestive system, by the way. Okay, so that's the process of the digestive system. Now, next up, required practical. We've got two required practicals back to back here. We've got the enzymes required practical, and we've got the food tests. So the enzyme required practical. This is what you need to understand first. We have starch. If you add amylase to starch, so starch is a carbohydrate, amylase is a carbohydrase. So if you add amylase to starch, it breaks it down into simple sugars. The simple sugar here is called maltose. All right. You can test for starch. This is one of the food tests we're going to learn in a second, by adding iodine solution. If you add iodine to starch, it will change color. It will go from an orangey brown color to a blue-black color. Okay. So if we had a test tube of starch and we added iodine to it, it will be blue-black. Okay. Now imagine we add amylase to that. So test tube, starch inside, it's blue-black. We then add amylase to it. Remember what amylase does? It breaks down starch into sugars. It basically gets rid of the starch. What's going to happen is you're going to get a color change. It's going to go from being blue-black back to brown-orange because there's no more starch in it. Once it's all broken down, there's no more starch. And we're using that concept in this practical. So this practical is basically trying to find out how fast an enzyme works and what factors affect it. So remember, there's two factors, temperature, pH, both of which can affect how fast an enzyme is working. So what you do is you have your test tube, you put starch in it, you put it on a water bath if you're testing temperature, for example, to warm it up. Then you add your amylase, and once that's happening, you take drops out of the test tube and you put it into something called a spotting tile. It has loads of these wells in it. So you put a sample. So imagine getting a sample from the test tube and putting it in every 5 or 10 seconds. So remember I said you've got a test tube at the start, and all it has in it is starch. We put iodine in it. When you put starch and iodine together, it makes it blue-black. And then we add amylase to it. Amylase is going to cause that starch to break down. So it's going to turn from being blue-black to orange. But it's not going to happen straight away. So if you get this mixture, at the beginning, it's just blue-black. So when you drop these at the beginning, you're just going to get blue-black wells. Blue-black, blue-black, blue-black. So 5 seconds, 10 seconds, 15 seconds, 20 seconds, 25 seconds. But then suddenly the amylase is going to break down all that starch, change it into maltose. Now you don't have any starch, so it started to go orange. So that means it's taken about 25 seconds. If we're making these drops every 5 seconds, 5, 10, 20, Sorry, 0, 5, 10, 20, 25. After 25 seconds, it's broken down into maltose. It's now orange. Now what you do is you repeat that process but at a different temperature. So imagine we put it at 100 degrees and the enzyme denatured. What's going to happen is you're going to get blue-black solutions the whole time. It is never going to turn orange because that amylase has denatured and it's never breaking down your starch. So it's blue-black the whole time. So if your enzyme has denatured, you're not going to get any orange wells here. The quicker it turns orange, the faster the rate of reaction, the faster the enzyme has worked. And that's what this practical is all about. That's what all of this describes. Okay, so the quicker it changes color from blue-black to brown-orange, the faster. And the point at which it happens the quickest, you can say that's the optimum temperature. Okay. You might have done it the other way around because this is where people get confused, because when you first add the orange brown, it turns blue-black, but then when it breaks down the starch into maltose, it turns orange-brown again. Okay. So that's how you test it with pH. So you keep doing it until it no longer turns blue-black. So let me repeat again because people are confused. So at the beginning, you've got starch, you've got iodine, you've got amylase. So it hasn't broken down. It hasn't broken down yet. So you have starch. Then eventually the enzyme is going to work and it's going to break down the starch into maltose. So now it's going to turn from blue-black to orange. And at that point, it's worked. The longer it turns, the longer it takes for the color to change, the slower the reaction is. It denatures. If it's denatured, it's never going to change color. Okay. Buffer solution is something you can use to change the pH. So remember, there's two factors we're investigating. Here we're investigating temperature, but you can also investigate different pHs as well. For that, instead of heating up the solution, you can just use a buffer solution to alter the pH here. All right. Okay. Next one, probably the easiest required practical. You're going to wish this is the one that comes up because it's just food tests. So there's 3/4 food tests you need to know about. First one is Benedict's reagent. Benedict's reagent tests for sugars. So if you have a test tube of sugars and you want to prove it's a sugar, you add Benedict's reagent. And what will happen is it will start off blue and then it will change color. There's no one correct color it changes. It could be green, it could be yellow, it could be brick red. If it changes color, it means sugars are present in it. For this one, you need a water bath, and that water bath needs to be around 75 degrees C. So you need to warm it up whenever you're using Benedict's reagent. The other ones, you don't need to do that. For starch, I've mentioned this already, you can use iodine solution and it goes from brown orange to blue black. For proteins, you use something called the Bioenergetics test. The color changes from blue to purple. Blue to purple. If it changes from blue to purple, it means protein is present. Then we've got a test for fats. And there's two here, guys. You probably learned the emulsion test in school. Can someone tell me how does the emulsion test work? Well, I go through Sudan 3. So Sudan 3 is another one you might have not come across in school. I don't know. It's another one that comes up in textbooks as well. You just add it to the solution. And if there's like a bright red layer that forms, it means fats are present. For emulsion, there we go. People are telling me you add ethanol. Let me add it here. Add ethanol and shake it. You shake the test tube. And if it goes cloudy, it means that lipids are present in it. So People asking, is it for AQA? Technically, I have never seen it come up in a question, and in the spec, it doesn't say it. Look what it says in the spec. It says carbohydrates. No, sorry. It says Benedict's. It says Biuret and iodine test. It doesn't say Sudan 3 or emulsion tests, but it does say lipids, which is why I'm telling you both of them. It doesn't specifically say Sudan or the emulsion test. That's why I'm teaching you both right now. Okay, it is odd, yeah. And this is literally what it says in the AQA specification. It's not clear on that, but I've never seen it come up. I've never seen either come up before. All right. Next thing we're doing, guys, this is circulatory system. A big topic here, a lot of memory tricks here as well. No memory trick for food tests unless anyone has one, drop it in the chat. I do have a lot for the circulatory system. The first one is the components of blood. So blood is one of the components in the circulatory system. The whole point of the circulatory system is to transport things around the body. All right. So the circulatory system is, the blood is made up of 4 different components: red blood cells, white blood cells, platelets, and plasma. And I've got memory tricks on how to memorize how each of these do their function. Red blood cells, they have 2 O's here for blood. That's basically, you can think of that as O2, because they carry O2 around the body. They contain something called hemoglobin, which binds to oxygen. So the 2 O's in blood, help you remember, is O2, because red blood cells carry O2 around the body, oxygen around the body. They contain hemoglobin, which binds to the O2, the oxygen. They have no nucleus, because it gives them more area to hold oxygen. And they have a biconcave shape. Biconcave shape, you can think of it as like a donut without a hole. It just has a dimple instead of a hole. And that increases the surface area. The surface area of it increases, meaning it can carry more oxygen. Now, people might think, but white blood cells also has 2 O's in it. Doesn't that mean it carries O2? No, because white blood cells has this T in it. Imagine rotating that T, making a cross. It doesn't carry oxygen. No, it doesn't. Not oxygen. Instead, what does it have? It has an I in it for the word infection. White blood cells fight infection. It has an I in it. This is the only one with an I in it. That reminds you that it fights infection. White rhymes with fight. It fights infection. Okay? How does it fight infection? We'll learn that in the next topic. There's 3 ways that it does that, and I have a memory trick for that as well. That's topic 3, fighting infections. Platelets. How do we remember this? Look at the letters that I've made a capital. We've got an S, we've got an E, we've got A, we've got an L. What does it do? It seals wounds. Platelets are there to give you scabs. If you get a cut, platelets seal your wounds so that blood can't escape, and at the same time bacteria and other microorganisms can't enter the body as well. They're fragments of cells. Yes, they're little pieces of cells. They're very small, and they seal wounds. Then we've got plasma, has an M in it for movement, because they move everything in the blood. It's like a liquid, and the liquid just carries all the red blood cells, the white blood cells, the platelets, and everything else you find in blood. What other things do we find in blood, guys? CO2, hormones, glucose. There's loads of things that we find in blood. It carries all of that. Plasma is there for movement. Exactly. There we go. All right. There we go. Now, That's the composition of blood. 4 main components. I've just summarized that table for you. Next thing you need to know about the circulatory system is the heart, the labels on the heart. Who knows my memory trick here? That's probably the one that's the most known, I reckon. The order that blood moves in the heart. We've got the arrows here. In through the vena cava, through the right atrium, ventricle, up the pulmonary artery. R valve, close, not R valve, not R valve. I've got it here somewhere. R valve, valve, L. So let me write down what each of these represent. Let me know in the chat if you do know. Write down what you think each word represents. So that each letter represents. Sorry. So this is for the right hand side of the heart. The L is for the left hand side of the heart. So blood comes in through the V first. The V. So that is the vena cava. On the right hand side, vena cava. So I'll show you here the diagram of the heart. Look at that. It's coming in through the vena cava. Then we've got the A for the right atrium. Then we've got the V for the right ventricle. And then we've got A for pulmonary artery. That's the order that it moves in. So vena cava, atrium. I'm going to put right because it's on the R side. Then we've got ventricle, also the right hand side. One thing you need to remember on diagrams of the heart is that the left side is on the right side of the diagram and the right side is on the left side of the diagram. And finally, we've got the artery, which is known as the pulmonary artery. That word pulmonary means it's connected to the lungs. Okay, think of it like that. Because once the blood comes out of the pulmonary artery, it goes to the lungs to get oxygenated. That's where all the alveoli are. And once it's taken, that oxygen, it returns back to the blood again. Back to the heart again, sorry. This time it comes in through the vein that's connected to the lungs, which is known as the pulmonary vein. Then through the atrium again. This time we're talking about the left atrium. Then we've got the left ventricle. Again, left ventricle. And then we've got out of the aorta. And then once it comes out of the aorta, it goes to the rest of your body, to all of the cells in your body, and it returns back round to the vena cava. That is the whole circulatory system. That's the root of the whole circulatory system. Frozen's got it. Yes, FRL classes member. So you definitely know from our classes. Okay, that's good. That's good. That's good. So we've gone through that as well. So we have a double circulatory system in our body. Okay, that means it's made up of 2 loops. This is one of the loops. It goes to the lungs and back. And then the second one is it goes to the rest of the body and back. That's why we've got a double circulatory system. And what you'll notice is the left-hand side of the heart is thicker than the right-hand side of the heart. It's got thicker muscles. That's because the blood needs to flow to the rest of your body, everywhere in your body, your head, your toes, your fingers, all your cells. So it needs to be pumped at a very high pressure. It needs to come out very fast, basically. On the other side, on the right-hand side of the heart, remember right and left are switched in these diagrams, it only needs to go to the lungs. And think about where your lungs are. Your heart is in your chest, your lungs are also in your chest. So it doesn't need to travel that far. So the pressure doesn't need to be as high there. That's why the left side is thicker than the right-hand side. The other thing is that the right atrium has something called a pacemaker in it. A pacemaker, which is basically a group of cells which control your heartbeat. Your heartbeat needs to pump at a very specific pace in order to pump blood around at the right, efficiently and at the right tempo. This sometimes doesn't work, specifically for older people. They have a pacemaker that doesn't work correctly, and that causes an issue where they basically need an artificial pacemaker, which is just a device with a wire in it, which attaches to the right atrium, which helps them to pump blood. The last thing in the circulatory system, actually second to last thing, is the 3 different types of blood vessels that you need to know. We've got that down here: veins, arteries, and capillaries. So 3 different types of blood vessels you need to know about. Memory trick here: veins carry blood into the heart. So that red in is to tell you that it carries blood into the heart. They have a V to remind you that they also have valves in it. Okay, valves are these things that prevent backflow of blood. So here is a vein, and you can see they've got these little gates. They're kind of gates. They only allow blood to flow in one direction. It prevents blood from going in the other direction. Another thing about veins is that they have a very large lumen compared to their walls. Their walls are very thin because the blood is traveling at a very, very low pressure. It doesn't travel very fast. Now, arteries are pretty much the opposite of veins. They carry blood away from the heart. That's what the A is for, away. They're pretty much the opposite. So remember what we said here? We said large lumen for veins and also thin walls. But for arteries, you guys see that T there? That T, rotate that and you get a cross, right? So imagine a cross. Doesn't have a large lumen. It doesn't have thin walls. It has thick walls and a small lumen. And I'm just going to show you by doing this. That cross is basically telling you it does the opposite. The reason why, so let's look at that diagram. The reason why is because it needs to carry blood at a very high pressure. The arteries, they take blood away from the heart, so they just come out of the heart, so they're going to be coming out at a very fast pace. We need to withstand that pace, so they need very thick walls so that they don't break. That's why they've got that. Then finally, we've got capillaries. Capillaries, they connect arteries to veins. So it goes arteries, capillaries, then veins. This is the best diagram to show you guys. Blood goes in, goes through the capillaries to all your cells, then it gets deoxygenated, comes back out of your veins. So that's what the C is there for. They are very thin. In fact, you can't see them with your naked eye. They have very, very thin walls, which are 1 cell thick and are permeable. They're permeable, which means they can pass substances through them because they need to go to all the cells in your body and give them oxygen, take carbon dioxide from them. So they need to be very thin in order to do that. Thick wall muscles, elastic. Yes, elastic, 1 cell thick, small lumen, permeable. Exactly. Excellent, guys. So those are the 3 different blood vessels that you need to know about as well. So we talked about composition of blood. We've talked about the structure of the heart, the order that blood flows through in the heart, and also the blood vessels that you need to know about. Last thing, which barely gets asked, is about the lungs. So let me quickly go through that. When you breathe in, air goes in through your trachea. It splits up at the bronchi, so 2 tubes that come off from the trachea, and then it branches off into bronchioles. So trachea, bronchi, bronchioles are the smaller tubes. And then eventually they go to the alveoli, which we've talked about already. The alveoli, remember, they're well ventilated. They have a large surface area, good blood supply as well, and thin walls as well to help you oxygenate that blood before it goes back to the heart as well. That whole process is called gas exchange. Aorta is a type of artery, correct? That is because it's going away from the heart. Blood is going away from the heart. non-communicable diseases soon. Actually, yeah, it's the end of this topic. We'll see how time goes. Yes, surrounded by capillaries for good blood supply. Excellent. Cardiovascular disease. This is when your heart doesn't work properly. It's issues with the circulatory system. The most common cardiovascular disease is coronary heart disease. So we've talked about all these veins and arteries. We've talked about the structure of the heart. There's one we haven't mentioned, which is the coronary artery. The reason why is because the coronary artery isn't inside the heart. It's on the outside of the heart. People always get this confused. The coronary arteries are arteries that you find on the outside of the heart, and they supply the heart muscles with oxygen so that they can contract. Remember, the whole point of the heart is to contract, to pump blood around the body. This coronary artery does that. But there's an issue. This coronary artery can get blocked. If you have too many fatty foods, too much oily foods, what can happen is you can get these fatty deposits on the coronary artery. And what that does is it closes it. It makes it less narrow. And eventually, if it gets completely blocked, you're now not getting any oxygen to these heart muscles. They're not going to be able to contract. And that's basically when you have a heart attack. This can also happen in the brain as well. Fatty deposits, that's when you have a stroke. So this is an issue that you need to know about and how we treat this issue. So one thing that we can use is a stent. A stent is basically this mesh tube that we use. We put it into the coronary artery and then we basically expand it. And when we expand it, it opens up the artery. So now this artery is fully open and blood can freely flow through that. That's quite a good thing, but the only thing is it requires surgery. Anything with surgery, there's always a risk with infection and blood clots forming as well. So you need to basically talk about the advantages and disadvantages of these. The second thing is statins. Statins are a drug that someone can take. If they have CHD, they can take statins and they lower your cholesterol. And when they lower your cholesterol, that means there's a reduced chance of these fatty deposits forming, meaning you're less likely to get CHD. Disadvantages of statins is the medication. You need to remember to take them every day. Otherwise, you're going to have the same problem crop up again. It's not very convenient having to take something every day. And because they're a drug, they could have side effects as well. Drugs tend to have side effects. The third thing, so there's four that we need to know. Third thing is artificial hearts. So if someone's heart has failed, what they need is a heart transplant. But the thing is, it's very hard to find donors for hearts. It's not usually available straight away. So a temporary solution is to give the person a temporary artificial heart, which does the job of the heart. It pumps the blood around the body. Now, the issue with this is that, again, it needs surgery. So there could be bleeding, there could be infection. It's not permanent, but a good thing about it is that it's not another person's heart. So there's no risk of rejection. When you're putting something mechanical in, something man-made, there's no risk of rejection. Your body's not going to try and fight against it. It's not going to produce all these white blood cells to try and kill it. And the last example you need to know is valve replacement therapy, which are when your valves, remember the valves are the things that stop backflow of blood. When they stop working, when they fail, you need to replace them. There's two things we can do. We can either replace them with biological valves, which come from like another mammal. They can come from cows or pigs, or mechanical valves. Both have advantages and disadvantages. Both have significant surgery. So you still get that risk of getting infections because it requires the surgery. Biological one, there could be rejection. Mechanical one, there won't probably be rejection, but the biological ones probably last longer. Okay. Yeah, Dennis, that's right. I would wear goggles Always to be safe. If they ask for safety precautions, I always say mention goggles. If you don't know what to say goggles. Yeah. What does the pacemaker do again? It controls the heartbeat. It's these group of cells in your right atrium, and it basically controls your heartbeat because you know your heart needs to go boom, boom, boom. It needs that rhythm. It can't if the rhythm isn't right, blood is going to flow the wrong way in the heart, and that's not going to be good. Cool. Non-communicable diseases. Someone asked me about this. To be honest, there is theory here, but a lot of it is data questions. Questions you get on this is data questions where they give you stats and data from a group of people, and you need to analyze it. But let me go through the basis of what it's trying to say. So health. What is health? It's a state of complete physical and mental well-being. You have physical health, you have mental health as well. Disease is anything that disrupts health. Okay, and we can have two different types of disease: communicable, which we're going to talk about in the next topic; non-communicable, which is this very small topic we need to know about. Communicable are ones which can spread between people. So something that you can pass on to someone else. There's like 7 that you need to know for topic 3. Non-communicable are ones you can't spread. Things like cancer, diabetes, type 1 diabetes. You can't spread those. So if you have it, you just have it. It's not spread by pathogens, microorganisms. That's what we're saying here. We've got examples: measles, salmonella, and malaria. They're all examples of communicable diseases, and we're going to cover that in the next session. What other information do we have here? We say diseases can interact. Okay. So that basically means if you have one disease, it can cause another disease. So for example, if you had a disease that causes you to have a defected immune system, so your immune system is not working properly, then that can lead to other diseases. It can trigger cancers. You can have some pathogens that cause allergies because of an immune reaction. It's basically saying one disease can lead to another disease. Health and lifestyle. So in order to avoid these communicable diseases, you need to have a balanced diet, reduce your stress, and also your life situation can also affect it. If you don't have good access to medicines and healthy food, then you're generally going to be more likely to have a non-communicable disease. Alcohol and smoking can also increase your chances. We call it a risk factor. It can increase your chance of developing these diseases, but doing exercise can reduce your chances of doing that. Other things to mention here: financial situation. So you could get questions just asking you to compare data. If you have a general public which are poor, then it could mean they have an increased chance of getting non-communicable diseases. You've got to think about the NHS as well when you're answering questions on this. So if there's like if you reduce a disease in the public, then if that can be reduced purely by diet and exercise, it helps the NHS because it means the NHS doesn't need to treat as many people. So like if people have CHD, you can reduce their chances of the general public getting CHD by teaching them how to exercise or teaching them about the benefits of exercise and diet. And what that means is there's less money spent by the NHS. They don't need to spend money on stents and statins and all of the things that we talked about there. That's what the financial impact is talking about there. Risk factors. This is something that increases your chance of getting a disease. So smoking is a risk factor for cancer. If you have cancer, so if you smoke, you increase your chance of getting lung and mouth cancer. Obesity is a risk factor. It increases your chance of getting type 2 diabetes. Alcohol, drinking alcohol is also a risk factor. It increases your chances of getting liver disease. And carcinogens, these are things that cause cancer. You have carcinogens in smoking. X-rays are carcinogens as well. They increase your chances of getting cancer as well. So what is cancer? Cancer is the uncontrolled cell growth and division. So if you have cells which are dividing uncontrollably, it's cancer, and it's caused by tumors. So we call them tumors, those cells that reproduce uncontrollably. Two types of tumors you can have: benign and malignant. Benign means it's not cancerous. Those are good tumors. The bad tumors are malignant tumors, which are cancerous, which means they can spread to the rest of the body. They'll start attacking other organs, and that's when you have cancer. So you just need to know, how can you reduce your chances of getting these non-communicable diseases, specifically cancer? So like I said, smoking, not smoking, sorry, not drinking alcohol, diet and exercise, controlling your weight, making sure you're not obese. UV exposure is one for cancer as well. And also there's some viral infections that can increase your chances of getting cancer as well. Okay. Yes, herd immunity. We'll talk about that in the next session. Someone said they're hungry, they want lunch. Let's see what we've got left. We still got plants to go through. We're a bit behind, guys. We're a bit behind. But this is the longest topic, so I don't mind that. What we can do then, let me make a mark of where we are right now. We're just about to do plants. This is where we are. So I'm going to teach some triple stuff now then, because we said that lunch was now. So for an hour, I did say now, right? Let me check the schedule again. I forget the times. Yeah, 12:15. All right, cool. So combined, guys, you guys can go have your lunch. We'll be back in an hour. Triple, guys, stay behind. We're going to go through culturing microorganisms now, which isn't long, so you won't be there for long. We'll go through xylem and phloem. Don't worry, next session we'll be there. Yes, I'll be quick. So combined, guys, I'll see you in an hour. We'll meet back at 1:15 here. Triple, guys, bear with me. This is a short one. It's a short one. So this is back to topic 1, to culturing microorganisms. And this is actually a required practical as well. So it is pretty important for triple people. So culturing microorganisms. First thing you need to know is how bacteria cells reproduce. Bacteria cells reproduce by a process called binary fission. Okay, so binary fission is literally one bacteria cell, pop, two bacteria cells. Okay, similar to mitosis, it produces identical clones. So it's asexual reproduction, basically. When these bacteria cells have enough nutrients and they're at a good temperature, a warm temperature, they can reproduce quite quickly, every 20 minutes. So they could ask you questions on working out how many bacteria cells are formed after a certain amount of time. So let's say we have a bacteria cell with a mean division time of 20 minutes. So that means every 20 minutes it divides. How many bacteria cells will be present after 3 hours? So what you need to do is work out how many 20 minutes there are in 3 hours. Every 20 minutes is splitting. So you start off with 1, splits into 2 after 20 minutes, then it splits into 4 after another 20 minutes. So in 3 hours, we have, in 1 hour, we've got 3 20s, right? So we basically got 3 times 3. So it's 9 times that it's dividing. So in 20 minutes, there are 9 divisions. Now we need to basically times 1 by 2 9 times, because it's dividing by, it's multiplying by 2 every time. So after 20 minutes, it's going to be 1 times 2. So you're going to have 2 bacteria cells. After 40 minutes, it's going to be 1 times 2 times 2. That's 4 bacteria cells. We want to do this 9 times, 9 divisions. So we're basically doing 1 times 2 to the power of 9. And that will give you 512. That's just an example of a question that you could get. An answer, yes, well done. And yes, it's asexual. Yes, yes, yes. Okay, now, growing bacteria in the lab. So to do this, we use agar jelly and a Petri dish. we use an inoculating loop. This is this metal loop that we've got, and that's how we transfer the bacteria to the agar gel. The temperature we use, remember I said the bacteria reproduce at 25 degrees warm temperature. So we use around 25 degrees and that causes them to multiply. That's how we grow bacteria in the lab. Now, one important thing when you're doing this, you need to make sure there's no other bacteria there. If you're trying to view a certain type of bacteria, you need to make sure there's no other types of bacteria in the agar jelly. Otherwise, that's going to affect your results. So you need to make sure they're uncontaminated. So to do that, we use these aseptic techniques. That's what it's called. Aseptic techniques are basically techniques that prevent other types of bacteria from interfering from your results. So what we need to do is we need to sterilize it. Sterilizing makes sure that there's no other bacteria present in the agar jelly. And we can do that by heating them up to very high temperatures. A lot of heat will kill a lot of bacteria. Then we also need to kill any bacteria we have in those inoculating loops, because remember, we're going to use that to transfer the bacteria onto the agar jelly. We can use that by passing them through a blue flame. Once we have put the lid on the Petri dish, we need to sellotape it. We need to use adhesive tape that prevents anything from entering. And also we store it upside down. The reason we store it upside down is because sometimes you get condensation on the top of the lid and that drips down onto the bacteria and it can affect your results. By storing them upside down, it prevents that from happening. So we don't heat them to high temperatures. The reason we use 25 degrees in schools is because if they're a bit higher, if they're around like 30 degrees, it can produce bacteria that thrive. Like if it's 37 degrees, they can produce bacteria that thrive in our bodies, which can be harmful to us. So we have to make sure we use a temperature which is lower. We use 25 degrees. Is Punnett Square spare paper too? Yes, it is. It will get enough air. They will get enough air. That's a good question though, but they should still have enough to grow. Would that be with aseptic techniques? These are all aseptic techniques, and a bit of both. So yeah, the temperature, that would be more safety rather than aseptic techniques. That's a good point that you made there. But you can still mention it if they ask you for a question on that. You can still say it's an aseptic technique. So the investigation that you're usually doing is you're investigating antibiotics and antiseptics. These are things that kill bacteria. All right. So if you have a Petri dish with bacteria on it, you can kill it using antibiotics. So what we do is we get these tiny circles, these paper discs, and we dip them in antibiotics and we put them on the bacteria and it kills the bacteria. And it basically diffuses outwards like a circle. So look at this. We've got bacteria. The yellow part is all bacteria. And we've got these little paper discs with different antibiotics on it. This one is a control. This one has nothing on it. It's just water, maybe. The other ones have antibiotics on it. And you can see there's like a clear section here. This represents all the bacteria being killed. And they're always being killed in this like circular shape. So we can estimate which one's the better one by looking at the area. The bigger the area, the more effective the antibiotic is. So you can see this one is clearly the best antibiotic because it's got the biggest area, the inhibition zone we talk about, we say, the biggest inhibition zone where there's no bacteria growing in it. Sometimes they ask you to do calculations here, and this is where you need to use your math skills. Area of a circle. For an area of a circle, we use pi r squared. So you would have to use a ruler, or they might give you the radius or diameter yourself. So if this is 44 millimeters, the diameter of this, you would have to half it to 22 to give you the radius. And then we have to use pi r squared there. So pi r squared, we're doing pi times 22 squared. Put that in your calculator and you'll get the area of your inhibition zone. So if they ask you for a calculation, that's the one that's most likely to get asked. All right. Yeah, that's what I wanted to go through there. There is more triple stuff, monoclonal antibodies. Do you guys want to do that now, or do you want to have the break and have a shorter break next time? Let me know in the chat. I'll do what the majority want. I can go through plant difference. No, I'll forget that. We can't do it now because we haven't done infection responses. So there's no point doing monoclonal antibodies now. I'll give you guys a lunch now until 1:15, and we'll do that next triple bit at the next break. That's much. Indeed. Wheel is for spinning the wheel is for the free boot camps and the free cram classes. All the boot camps and all the cram classes are free. So if you haven't done that already, sign up. I'll put the link down in the chat again. And yes, you guys can have a break, not for an hour. We'll be back at 1:15. 1:15. 1:15, we'll be back. Okay? Yeah. Have a good rest, guys. Have a good lunch, and we'll be back energized for the final 2 topics. Hey guys, how's everyone's lunch? What do we all have for lunch? Let me know. Someone said samosas. Nice. Those of you who haven't, guys, we've got the cram class which you guys can sign up to. So remember, these are going to be the day before your exams, the evening before your exams. I know a lot of you have already signed up, but for those of you who just came, you can sign up, and I'll put the link in the chat right now. Pasta. Someone had pasta? Sandwich. Nice. Yeah, same here. I had a sandwich, had a small sandwich. And I hope you guys are re-energized. We have second half to go. We're carrying on with the organization stuff. So we stopped at plant cells and tissues. You rewatched? Okay, that's good. That's good. How long is the cram classes? I think it's like 5:30 to 9 or something like that. Double check. They're very intense because we're literally cramming everything in the night before. But we've had a lot of positive feedback from it. It's good for your revision, like the day before an exam when you're trying to get all that information in. That's mainly what it's useful for. So even to have in the background while you're revising, doing past papers, it's good to do. Right, so plant cells and tissues is next. We have 6 different tissues that you need to know about. We've got 2 here. I know it's 5 bullet points, but 2 of them are xylem and phloem. We've got the epidermal tissue, which is the very top and at the bottom of the leaf. We've got the palisade mesophyll tissue, spongy mesophyll, xylem, phloem, and meristem, which we've already talked about. That's the tips of shoots and roots. And remember, that's what type of cells do you find on meristem tissue? Let me know in the chat, guys. I'll show you guys this diagram. Where do you find meristem tissue? Stem cells. Yes, Phil, straight in. And blue G8's got that as well. So epidermal tissue, like I said, it's at the very top. We call it the upper epidermis, but we also have the lower epidermis. It's the top and the bottom. Upper epidermis is completely transparent. It lets light through to the palisade mesophyll layer. Palisade mesophyll layer is a layer with several cells with very concentrated chloroplasts. So it has a very high concentration of chloroplasts in them because that's where most photosynthesis happens, in the palisade mesophyll layer. So the light goes through the transparent layer of the upper epidermis into the palisade mesophyll. And that's why it's near the top as well, because it can receive maximum sunlight for photosynthesis. Underneath that, we have the spongy mesophyll. So here you can see the cells are quite spread out. There's a lot of air gaps in the middle. All of these gaps here, that's for air. Because what happens there is gas exchange. You need carbon dioxide to go in. You need oxygen to get out during photosynthesis. So that just helps the diffusion process. Then we've got the xylem and the phloem, which we're going to talk about in a second. And underneath that, we've got the lower epidermis, which has the guard cells and the stomata. The stomata are holes that you find at the bottom of the leaf. You get loads of stomata at the bottom. And guard cells are basically cells that open and close to open and close the stomata. All right, so more info on that in a second. The organs that you need to know in a lot of detail then, the phloem and the xylem. So here we've got a memory trick, phloem and xylem. And if you look, you can see that I've written phloem with a tick here, a backwards tick, a green tick, and a cross for xylem. And I've written the word let. Someone mentioned this earlier in the stream. Does anyone know what the let stands for? So this is a memory trick to remind you what happens or what's the differences between the xylem and the phloem. So L, anyone know? Anyone know? I'll wait for you guys to type. While you do that, I'll type this first one. First one is living cells. L is for living cells. Yes. E, thank you, Blue. And E is for end walls. And T is for two-way flow. Okay, so I'm going to use this to help you memorize all the differences between these. So for phloem, the phloem is made up of living cells. The tick tells you everything in let is true. It's made up of living cells. it has end walls in it. So if you look at the structure, can you guys see here? In between each cell, this is a cell. It's got these walls in it with holes in them. It has end walls. But xylem doesn't have that. Xylem doesn't have end walls. So it has a tick there. It has two-way flow. Whatever it carries, it can go in both ways. It can carry substances up and down. The phloem is used to carry sugars. It transports, I'm going to say transports. It transports sugars around the plant, both up and down. It's got two-way flow. So all of this have ticks. Xylem, the X is a cross. It's not made up of living cells. It doesn't have end walls. It doesn't have two-way flow. So it's not made out of living cells. It's made out of dead cells. And those dead cells, because dead cells aren't very strong, they're strengthened by a substance called lignin. End walls, they don't have end walls in them. They're completely hollow. They're completely hollow. And they don't have two-way flow. They only have one-way flow, which is upwards. So water is what's carried in the xylem tube, and it only travels up the plant. It never goes down the plant. Okay, so you can use let to remind you of those. There we go. Everyone's saying the right stuff there. I'm seeing all the correct things. Well done, guys. Then we've got root hair cells. We don't need to talk about root hair cells again. We've talked about it twice/3 times already. Stomata, we do need to talk about a bit more. So like I said, they're the holes that you find in the middle of the leaves. Not middle, sorry, the bottom of the leaves. And they're controlled by these guard cells. So either side of the stomata, you have these guard cells. Now, the guard cells can open and close, but that depends on how much water they have within them. So if they're full of water, we say that they're turgid. And that causes them to open. That happens in high sunlight conditions. If there's a very high light intensity, these guard cells get filled up with water and they open up. So when they open up, they have a lot of carbon dioxide going in, a lot of oxygen leaving, a lot of gas exchange. That's because when there's a lot of sunlight, it wants to photosynthesize more. The plant wants to photosynthesize. It wants to make use of that sunlight. So it opens up the stomata. At nighttime, it does the opposite. It becomes flaccid. It gets all that water leaves these guard cells and it closes. And that's because there's no sunlight for photosynthesis. What can happen to a plant, however, is it can lose water through those stomata. That's like a byproduct. It doesn't want to lose water all the time. It does keep it cool, but it doesn't want to lose water. That process is called transpiration. So in order to minimize water loss at nighttime, it closes by becoming flaccid. Okay, so what does the T stand for in let? It stands for two-way flow. Two-way flow. Yeah. Now, we're going to talk about that root, the xylem root. You don't need to know the phloem root. You need to know the xylem root. So the phloem root, we call that translocation. That's the process of sugars moving around the plant. For water and minerals, we call that transpiration. The route that it follows is called the transpiration stream. And you need to know the route, the order of the route. Where does it start? Where does it end? So here we have a memory trick here where we start off with the R. So water always goes into a plant in the roots. So this is rooksets. The R is for roots. It goes into the roots. The water goes into the roots by osmosis. Let's write these down. So roots, it goes in by osmosis. It travels up the plant. Remember, it only travels up. It doesn't go down. It's one-way flow, not two-way flow. And it goes up through the xylem tube, the xylem vessel. Then, so it goes up, 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 the xylem, xylem, xylem. It reaches the leaves, and then there's the E. It evaporates. There's evaporation occurring. This whole process is called transpiration, and it occurs through the stomata. I think I've seen a 5 marker on this, asking you to describe the roots. If you write Roxettes and write the points, obviously write it in bullet points and put some context in it. Don't just write these words. Then you'll get all the marks there. What's the difference between transpiration and translocation? Translocation is the movement of sugars. It happens through the phloem. What I'm describing here is the transpiration stream, which is the route of water in the plants. Then you have transpiration, which I'm going to talk about now, which is when water gets lost from a plant by evaporation and diffusion. Okay, so transpiration, you can increase the rate of transpiration. Transpiration happens faster when these 4 factors are changed. So we can use HALT to remember this. Humidity, airflow, light intensity, and temperature. All of these make transpiration fast or slow. They affect the rate of transpiration. For so long, I used to call it thou as a memory trick. And then last year, someone told me to use HALT because that makes more sense, is actual word. So only last year did I change this to HALT. I used thou for such a long time and it just didn't make sense, but this is much better. So thank you, whoever it was that told me about this. Humidity. As humidity increases, so as humidity increases, transpiration decreases. Transpiration decreases. CO2 concentration is not a factor for this. No, that's for photosynthesis. This is for transpiration. If airflow rate increases, so that's basically wind. If it's a windy day, then transpiration increases. Let me just copy and paste this to make it a bit faster. Increases. It makes transpiration faster. That's basically because you're removing all the water from the outside of the leaf. You're increasing the concentration of gradient, and it makes diffusion of water faster. Light intensity. If you increase light intensity, transpiration also increases because remember, it opens up the stomata. It makes those guard cells turgid. It opens it up. So now water can leave through the stomata faster as well. And temperature, if you increase temperature, it causes transpiration to also increase because more evaporation happens. If you have hotter conditions, evaporation is going to happen faster. The water is going to turn into gas quicker. So as light intensity increases, why do the stomata become turgid when light intensity is high? That's just the way that they adapted. They just adapted to become turgid when the light intensity is high. There's one experiment. It's not a required practical. I've seen it once or twice. This setup, have you guys seen this before? It's called a potometer and is used to measure transpiration. It barely comes up, but I think I should just tell you just in case it does. So this is the setup here. You've got this container. It's got water in it, and it's connected to this beaker of water. And what you have here is a plant, and it's actually growing in the water. So this water level should be up to here. And what happens is water rises up, gets taken up by the plant, and then it evaporates. So transpiration occurs. And you can measure how fast transpiration is occurring by adding an air bubble here and seeing how fast it moves along. Because remember, the water is getting sucked up by the plant, right? So if an air bubble moves really quickly from left to right here, it means transpiration is fast. And if it's moving slowly, it means transpiration is slow, okay? So this is just something that could come up, but has barely come up in the past. You don't need a light bulb here, no, because that again, that's for photosynthesis. This is a different experiment. This is transpiration. Transpiration is the loss of water. Photosynthesis is when plants make glucose for food. No pondweed. No pondweed. We're going to do that in the 4th session, guys. The pondweed one, that is for photosynthesis. This is purely for transpiration. Okay, cool. Topic to done. Topic 3, infection and response. Let's get into that. First thing we're going to talk about are the 4 different types of pathogens. So remember, we talked about diseases, 2 types of diseases you can have: communicable, non-communicable. We talked about non-communicable diseases already. We talked about cancer. Communicable diseases are diseases that can spread between organisms, and they're caused by pathogens. The definition of a pathogen is microorganism that can harm the body. And there's 4 types that you need to know about. Bacteria, so bacterial living cells, they produce toxins to make you feel ill. They're the only ones that produce toxins. Okay, remember that. Then we've got viruses. Viruses, they're not cells technically. They reproduce slightly differently to bacteria. So bacteria, they reproduce sort of like mitosis. Those of you who do triple know that bacteria reproduce by binary fission. But you literally have one bacteria cell, it splits into 2. Okay, viruses, they reproduce very differently. So here's a virus. It attaches to the end of a cell. It injects DNA into it, and it basically instructs the cell to make copies of the virus. So we've got copies of the virus here. And that cell basically gets filled up with so many viruses that the cell bursts, and then it releases the viruses to make you feel to spread to other cells. And that cell damage is what makes you feel ill when you've got a viral infection. Bacteria is different. Bacteria make you feel ill by producing these poisons called toxins. That's what makes you feel ill if you have a bacterial disease. So these are the 2 that get asked the most. The information gets asked most about bacteria and viruses in exam questions. But there's 2 more that you need to know about, which are protists and fungi. So protists are eukaryotic. Remember, eukaryotic, EU, they have a nucleus. They exist as parasites. There's only one protist disease that you need to know in your exams, and that's malaria. Malaria, which we'll cover in a second. And the way that they spread, different to both bacteria and viruses, they spread with a vector. So a vector is like a mosquito for malaria. A mosquito comes in, it takes your blood, and it takes that malaria protist as well. But the mosquito itself never gets malaria. It has the protist in it, but it doesn't get malaria. So it's technically a vector because it doesn't get ill with it. It carries it to another human, again, takes their blood and transfers the protist to that second human. Now that second human has got malaria, and the malaria has spread. But the mosquitoes are just spreading it. They're not getting the disease themselves. Therefore, they're known as vectors. We don't call them a carrier. It is carrying it, but we don't call it a carrier. No, there's none for A level. How are these diseases spread in animals and plants? We're going to talk about how they're spread as well. Someone's got a memory trick for food tests. Let's see. Oh, that one. Okay. I'll take a look at the end. Interesting. Okay, I'll take a look at the end and I'll see. Then we got fungi. Fungi can be multi-celled or single-celled. They have these structures called hyphae. If you've got like a fungal infection, so if this is your skin, the fungus will attach onto your skin, and then it'll basically produce these hyphae, which are like roots, and it takes nutrients from it. That's how it grows. And that's how it also reproduces. It produces spores, which are kind of like seeds that float around in the air, and then it goes to a different section of your body to another person as well. But they don't ask much on this, on hyphae, in fact. Okay, athlete's foot is fungal, yes. But if you're AQA, you don't need to know about athlete's foot, really. These are the 7 that you need to know, all right? So we've got 3 viral diseases here. Viral, that is measles, HIV, TMV, tobacco mosaic virus. We've only got 1 fungal one that we need to know about, 1 protist disease, and 2 bacterial diseases. Okay. For each of these, this basically summarizes all the stuff you need to know about them. You need to know how they're spread. So measles are spread through sneezing. It's a bit like COVID. Symptoms, you get a red skin rash and fever. Treatment, now this is important. This has come up. They've asked you what MMR stands for. You can treat it with a vaccination called MMR. It stands for measles, mumps, and rubella. That was I think it was a 1 marker in a question before. That's also a way that you can prevent it. Then we've got HIV, which is a sexually transmitted disease. Symptoms, flu-like symptoms, and it progresses to AIDS, where it basically breaks down your immune system. If you have no immune system, when you get infected with a disease, you can pretty much die. There's nothing fighting off that disease. Is COVID on the mark scheme now? You can use it as an example, but it's not one of the ones on the spec. You don't need to know about COVID in the spec, but you can use it. I'm sure it will be in the mark schemes if they ask you to name a viral disease or something like that. How can you treat HIV? You can use something called antiretroviral drugs. And to prevent it, safe sex, so using condoms, and also no needle sharing as well. Tobacco mosaic virus are plant viral infection. So the way that's spread is plant contact. So leaves touching other leaves can spread TMV. It leads to discolored leaves. And if in a plant the leaves are discolored, if they're yellow instead of green, that means they don't have a lot of chlorophyll in them. If they don't have chlorophyll, it means they can't really photosynthesize. If they can't photosynthesize, it means they can't grow. So it leads to stunted growth, less photosynthesis. There's not really a treatment for it. You just need to pretty much just kill the plant and make sure you don't touch another plant once you've touched one which is infected. Rose black spot is a fungal disease. It's also a plant one. It also leads to discolored leaves, but it does something weird. It forms purple/black spots on the leaves. Again, same thing. It stops or it reduces photosynthesis in a plant, which means it can't grow as much. This one spread through the wind and water, so it can spread through the soil. So if you want to remove it, get rid of it, you can remove the leaves with the purple spots on it. You can add fungicides. These are things that kill funguses. And you can use fungicide sprays as well. Malaria, I've talked to you guys about already, how it's spread using mosquitoes. Symptoms, fevers, chills, tiredness. You can use anti-malarial drugs. Prevention, I've seen this quite a few times. Mosquito control, so using mosquito nets and destroying where mosquitoes breed. This is what some countries do. They find their breeding locations in like swamps, and then they can destroy those so that they don't breed and mosquitoes then get released. You can also use insecticides to prevent them from coming close to you. Next, we've got salmonella, which is a bacterial disease. It's basically food poisoning. If you have food contaminated with it, usually poultry, so chicken and duck, then if it gets into your body, it can lead to fever, stomach cramps, vomiting, and diarrhea. And because it's bacterial, we can treat it with an antibiotic. We're going to talk about antibiotics in a second. Anything bacterial, so salmonella and gonorrhea, you can treat with antibiotics because they are bacterial diseases. How can you prevent it? Well, washing your hands when you're cooking. If you're suffering from salmonella, washing your hands and cooking your food thoroughly can help. Then we've got gonorrhea, which is another STD, a sexually transmitted disease. It's bacterial this time. It can lead to painful urination and genital discharge, again, treated with antibiotics, and barrier methods such as condoms can stop it from spreading. So you get questions on how pathogens can spread. You can mention through water, through the air, like sneezing, or direct contact. These are 3 things that can lead to pathogens spreading. You can reduce the spread of pathogens by good hygiene. So that's for like the salmonella, destroying vectors for malaria, and also isolating. That's what we did in COVID, right? So think about that if they ask you questions on how to prevent the spread of a disease. It can happen through water as well. It's just these are the ones that are mentioned in the spec. So I put the ones which are mentioned in the spec. Vaccination can help as well, but I'll go through that when we talk about vaccinations. No, do not base your revision on predictions, please, guys. Don't do that. They're wrong so many times. They're wrong. You don't know who to trust. Don't base it on predictions. That's why we never do predictions. We just do these boot camps, which cover everything, because we cover all things that can come up. Okay, so we've talked about diseases. We've talked about pathogens. Now we're going to talk about how our bodies prevent us from getting ill. We have several disease defenses. We have 5 that we need to know for our exams, and we use a memory trick, Snazzy, to remember them all. So S is for the skin. The skin is in the very outside of your body. It's basically a barrier, very useful. It also produces these oils, these antimicrobial substances, which kill bacteria that get onto it. So very, very useful. That's probably our most useful defense, because it prevents us from getting so many diseases. Now, if your skin gets bypassed, then we've got our nose. In our nose, we've got several defenses as well. We've got hairs and we've got mucus that gets produced. So the mucus traps pathogens or particles that contain pathogens, and so does the nose. So it stops it from getting further. You don't want these pathogens getting into your blood. If they get into your blood, then you're basically infected and you're ill. So the nose prevents it from going further in, because think about where the nose leads to. The nose leads to your air passages and your lungs. And you guys remember, your lungs have alveoli in them, which link to your blood. They've got blood capillaries. You don't want the pathogen to go through that whole route and end up in your blood. So even the next section, the air passages. If you guys remember when we did the lungs, we got the trachea and the bronchi. They've got their own defenses as well. They produce mucus. So that's your phlegm that you have. And that traps pathogens. And they have cells, special cells called cilia, which are like hairs. They're not hairs, though. Don't describe them as hairs in your exams. They're hair-like structures. So they're like hairs, but they can move. So when you get a lump of mucus with pathogens in it, the cilia basically wafts it back up to the top of your to the back of your throat, so it doesn't go further into your lungs, and you basically can swallow it, and it goes into the stomach. You would rather your pathogens go to your stomach than your lungs, because in your lungs you'll get infected, it'll go into the blood. But if it goes to your stomach, if you guys remember, has acid in it. It has hydrochloric acid in it, and that kills bacteria. So these are all the external defenses that we have before you get infected. But if you do eventually get infected, if that pathogen does end up in your blood, then your last line of defense is your immune system. So every time you get ill, it's your immune system that saves you from dying. That's what we're going to talk about now. Can I use these booklets for revision notes? Yes, because I literally went through every spec point. So use the revision booklets, try the exam questions, but then at the end, do full exam papers as well. Okay. What does the skin do again? It's a barrier, and it produces oils or antimicrobial substances. Yeah. Antibodies, antitoxins. I'm about to do that right now. Okay, so we're going to talk about the immune system in more detail. This is the thing you need to talk about in more detail. So this is everything that I've talked about now. We've got the immune system, which is basically made out of white blood cells, and it fights off infections and pathogens using 3 different methods. This is a memory trick for that, for how the immune system works. We use APA. So APA. First thing is antitoxins. So our immune system is made up of white blood cells. Some of those white blood cells are called lymphocytes. And lymphocytes produce antitoxins. Do you guys remember I talked about toxins earlier? I said bacteria produce toxins. Those are the poisons that make you feel ill. Well, our white blood cells fight back by producing antitoxins, which neutralize those toxins. That's one way. The second way is by phagocytosis. Phagocytosis. So this is this process here. So you can see we've got a white blood cell known as a phagocyte. And what that does is it goes up to a pathogen and it changes its shape. It looks like it's eating it, but it's not actually eating it. It's a cell. It doesn't have a mouth. It changes its shape to surround it, and then it digests it. So it completely destroys that pathogen. We say it engulfs it. That's the key word that you use there. So that's phagocytosis. The last way, which is probably the one that you need to know the most information about, is antibodies. So someone was asking, what's the difference between antitoxin and antibodies? Antitoxins neutralize toxins. Antibodies, they're there to attach onto something called antigens. Loads of antis here. So every pathogen has this structure on its outside, on the very outside surface, called antigens. All right. This is how a body identifies a pathogen. If it's seen that antigen before, it knows it's seen that infection before and it knows how to deal with it. But if it hasn't seen it, it basically needs to try and produce antibodies to attach onto the antigen. So look at this. This is a lymphocyte, a white blood cell. I know it's orange, but it's a white blood cell. And it produces antibodies, which are these Y-shaped things, which attach on to the antigens of the pathogen. Okay, so they all attach on there. These antibodies are basically just proteins. They're designed to bind to antigens of a pathogen. And what happens is once they've binded to several pathogens, it basically marks them for destruction. It's like a homing signal. It's telling the phagocyte, oh, there's a pathogen here, come and eat it. And then the phagocyte comes and destroys it. So that's what antibodies do. They act as a signal for these pathogens to be killed. So imagine this. Imagine you've just been. Imagine you've been infected with a disease and you've got several pathogens in your body. You're ill. Okay. Your body has never seen this infection before. So what it does is it tries to figure out what the correct antibody is, because, like I said, different pathogens have different antibodies, antigens. So there might be some which have a different shaped antigen. So there might be some which have like a rectangular shaped antigen, for example. We only have antibodies for the pathogens that we have already experienced in our life. So if you've had chickenpox before, you probably have chickenpox antibodies in your body. But if you come across something new that your body has never experienced, it's not going to recognize that antigen. So while you're ill, it's trying to figure it out. It's trying to find out what the correct antibody is. And hopefully, eventually, it will figure it out. And it'll start producing these antibodies. And it will kill all the pathogens in your body eventually. So that's why you get a cold for a few days. Your body is trying to figure out the correct antibody. It figures it out. It kills all the pathogens. And now you're better. Now, the issue is that cold could come back. So once all of those pathogens have just died out, once you've killed it, once you're better, there's no need to have all those lymphocytes there. So most of these lymphocytes just die off. We don't need those right now because the pathogen has died off. But there's a few which don't die. A few lymphocytes remain after the infection has happened. And we call those memory cells. Memory cells. So memory cells are there just in case this pathogen appears again. If that same pathogen re-enters your body again, now your body will recognize it. Those memory cells are there. It will say, right, these are the antibodies. Let's produce them. And they produce them very rapidly and a very large volume, more of them. So that means it kills off the pathogen before it's even had a chance to make you feel ill. So that is when we say a person is immune to a disease. All right. Let's see what these points are saying. So antibodies bind to the antigen. Yes, the body remembers them. Don't say they remember them. Just say memory cells. It recognizes it, yeah, fine. But the memory cells don't remember them. They just have the antibodies to destroy those pathogens instantly. And they produce the antibodies more quickly. That's an important point. And in a larger quantity. Do we have to talk about memory cells? Yes. Yes, you do need to mention memory cells if it's talking about someone, how someone is immune. Okay? So those are the three different things for that. Now, something external we can do to fight a disease. So we've talked about our natural defenses against disease. We have man-made defenses as well. Vaccination is something that we have. What a vaccination does is basically an injection which has the pathogen it's trying to protect you against, but it's dead or inactive. So if you want a vaccination for measles, you basically inject the person with measles, but it has a dead or inactive version of it. So it's not going to make you feel ill. But it does look like measles. So when it goes into your body, it still looks like a pathogen. So your body is going to think it's trying to attack it. So your body does what it usually does. It tries to produce antibodies to kill it. And it produces these antibodies and it keeps these memory cells. But in this whole time, you haven't gotten ill because this is a dead or inactive version of that pathogen. But if a real version of measles was to enter after, then your body will already have the antibodies, so rapidly produce those antibodies to eradicate it before you're even ill. That's how vaccinations work. So it's quite smart. It's kind of tricking your body into thinking you're ill so that you produce these antibodies so that you don't actually get ill. Congratulations, not 11K. Oh, we reached 11K. Oh, nice. I was waiting for that. Thank you. What about herd immunity? So herd immunity, I think we mentioned it here. So that's when you vaccinate a large population of people. Okay, so these are advantages and disadvantages of vaccinations. So they're good because it can prevent epidemics and pandemics with COVID. If you vaccinate enough people, a huge percentage of the population, then that pathogen is not going to go around as much because people are just eradicating it straight away. It's not dividing in their body load. So there's less of that pathogen floating around in the population. So that's very useful. It can eradicate a disease. Smallpox was one, polio nearly eradicated because we used vaccinations for those, and most of the population's got vaccinations for those. Disadvantages of vaccinations, they don't always work. They don't always guarantee immunity. Sometimes they make you stronger, like COVID, for example. It didn't prevent you from getting COVID. You still got it, but you just got a milder form of it. It was less lethal. And also you can get side effects from some vaccines as well. So you could get asked questions on advantages and disadvantages of that. Has dead or weakened pathogens? Yes. Memory lymphocytes and lymphocytes different? Yes. Memory lymphocytes or memory cells are just lymphocytes that haven't died off after an infection. So after infections, the lymphocytes that you used die off, but a few remain, they survive, and those are called memory cells or memory lymphocytes. So your body will always produce antibodies, but when would it produce antitoxins? It will only produce antitoxins if you've got a bacterial disease, because remember, only bacteria produce toxins. So there's no need to have antitoxins for viruses because they reproduce a whole different way. They make you feel ill a different way. Why were some people more affected by COVID than others? That's just because everyone's got different genes, different genetics. So people react to a disease differently. There's variation. That's more paper two stuff. But that's the reason why it wasn't lethal to everyone. Oh, that's great to hear, Safa. Thanks for that. What are phagocytes, Dennis? Let me say that again. So phagocytes are a type of white blood cell. They're the ones that engulf a pathogen. They're the ones that eat but we say engulf, and they digest them to get rid of them. Okay, so let's go through antibiotic and painkillers. So these are two different types of drugs that you need to know about, antibiotics and painkillers. So painkillers are things like paracetamol, ibuprofen, aspirin. All of these are painkillers. They don't actually kill the pathogens. So you take them to reduce the pain, to make you feel better, but they don't actually make you feel better. They don't actually get you better faster, because they don't kill the source of the disease. They don't target the underlying cause of the disease. They don't kill bacteria, viruses, protists, or fungi. What's more useful for you getting better is an antibiotic, because antibiotic kill bacteria. They only kill bacteria, okay? And what they do is they prevent them from reproducing, so it reduces how many bacteria you have in your body, meaning you have less toxins, you're not going to feel ill, and it's going to eradicate them faster. But they can only be used for bacteria, so that's a big problem. They're not effective against viruses. You need to know why. Because do you guys remember how viruses reproduce? Do you remember that diagram I showed you? I said that viruses reproduce inside cells, right? Like this. So in order to kill a virus, if you've got a viral infection, in order to kill a virus, you need to kill these cells as well. And these are healthy, these are human cells. So it's very difficult to kill a virus without destroying healthy human cells. So antibiotics can only be used against bacteria. Where are we? We are here. Yeah. So antibiotics can only be used against bacteria. Now, problem with antibiotics, a more recent problem, is resistance, antibiotic resistance. Okay, so this is kind of the evolution topic as well, but it does come up in paper 1. When you overuse antibiotics, so not a single person, if a population overuses antibiotics, there's a risk of resistance. So that's basically when it evolves to stop being affected by the antibiotics. The antibiotics are meant to kill them, but they evolve, because remember, bacteria, they reproduce very quickly. They reproduce every few minutes, every 20 minutes or so. So they can evolve and change very quickly. Their generations are much quicker. So they can very quickly become resistant to antibiotics. So if you use quite a lot of antibiotics, they can become resistant, and that means you can no longer kill that particular bacteria cell. So an example was MRSA happened. This was a bacteria that we used to have a cure for, which was an antibiotic, but because we overused it, it mutated and evolved into a strain which we couldn't kill. The antibiotics would no longer kill those. So that was a problem, because loads of people started dying because we couldn't use any sort of medicine to destroy it. We needed to develop new medicine to destroy it, but it takes years and years for us to develop new medicine. So it's very, very dangerous. Now, we've got a few procedures that we use to avoid this from happening. And one is doctors only give you antibiotics if you're really seriously ill. So if you just go to your GP for like a small cough, your GP will likely not give you antibiotics for it, because it's a waste, because a cough isn't affecting you too much, and you'll get better within a few days. Your immune system will fix it, right? So they avoid giving prescribing antibiotics. The other thing is, your doctor will tell you, have these antibiotics for 7 days, but even if you feel better after 2 days, carry on having the antibiotics. That's very important, because think about what's happening here. Let's say you've got 1,000 bacteria in your body and you're very ill. So you take antibiotics. The antibiotics on day 1 kills, how many did I say? We have 1,000. It kills 500. On day 2, it kills another 400. Day 3, it kills another 90. So you've got 10 bacteria left
Now you're going to start feeling better. So you're going to think you're cured, but you've actually got 10 bacteria left in your cells, in your body, and they are the strongest bacteria because they haven't been killed by the antibiotics, right? So they are the mutated ones. They're the ones that are better adapted. So if you don't carry on taking these antibiotics and overwhelming them, there's a chance that those are going to survive. Those strong ones are going to survive, and they're going to reproduce and produce even stronger, produce more copies of them. And that's more dangerous because these antibiotics are less effective. So this is why you need to complete your full course of antibiotics. Okay, so that's antibiotics. Now, other drugs you need to know about. I have been trying to think of a memory trick for this for ages. There's three drugs where you need to know what plant or no, what origin they come from. Okay, so we've got aspirin is one. Aspirin comes from willow bark. Aspirin is used as a painkiller and to lower fever. You need to memorize this. Another one is penicillin. Penicillin was discovered from mold. It was the first ever antibiotic that was discovered. And then we've got digitalis that comes from foxgloves. Okay, so these are three origins and three drugs that you need to know about. So if anyone can come up with a good memory trick here, let me know. AWPMDF. All right. So what is everyone saying? Does this mean they mutated? Yes, they mutate. Exactly. Mutations. UTI, I'm not sure how a UTI works. That is also probably bacteria as well. Do they produce identical offspring? Good question, Hydra. They produce identical offspring, so they don't have variations. But what they do is they mutate during their lifetime. So they change while they're alive. And then once they've mutated and they replicate, you're going to replicate the mutation as well. It's a good question. Is the use of antibiotics to kill bacteria? Exactly that, Dennis. Antibiotics are used to kill bacteria. Padmore. Padmore. Does that work? What's the A for? What's the difference between virus and viral infection? It's all the same. Viral means it comes from a virus. So a viral infection is it's when you get ill with a virus. Could you say bacteria undergo natural selection? Technically, yeah. Because the one which are better adapted survive. So you could say that as well. All right. Cool. If you do find a memory trick, guys, let me know in the chat. I love hearing these memory tricks. Oh, okay. Last one. Nice. Nearly finished early. So, discovery and development of drugs. So, whenever you create these new drugs, any new drug that's being created, before you release it to the public, there's a series of stages you need to do. You need to test it thoroughly. You need to make sure it's safe for the public and it works. And you need to know the different stages that occur. So we have another memory trick here, which is CAVIP. CA, the first two stages of testing a drug, is called preclinical tests or preclinical trials. Second is clinical trials. So when you first have a drug you want to test, you test it on cells and tissues. Cells and tissues. So these are human cells, human tissues. We test the drug to see if it's harming them or anything like that. Okay, but that's not enough. We need to test it on full systems, organ systems. We can't just test it on tissues and cells and be like, right, it's not harming the cells or tissues. We can just give this to people. We can give it to humans now. We need to test this on full systems. Like, does it affect your blood pressure? We can't test that with its cells or tissues. We need the whole circulatory system to do that. So this is where we use animals, live animals, to do this. So we test the drug on live animals next. Now, we test them for three things. These are three scientific terms that you need to know. Efficacy is the first one. That means, does the drug work? How well does it work? Is it doing its job? Efficacy. Then we've got toxicity. Does it harm the animal? Is it going to harm humans? And last one is dosage. How much should a human be taking and how often should they be taking it? So 3 things that they ask you: efficacy, toxicity, and dosage. So they test it on these live animals. And once they've done that, they move on to clinical trials where they test on humans, real humans. The first humans they test it on are volunteers, human volunteers. These are healthy individuals. So these are people that don't have the disease. The reason they do that is because they want to test for side effects. So they give them a very low dose at the beginning of this drug, see how they react, and then they increase it slowly and slowly and slowly and slowly. And they find the optimal dose, which produces the least amount of side effects. And they go with that. And from there, they decide what the dosage of a person should be. And then that leads on to the last stage, where you give it to the eye, which is for ill patients. So these are people with the disease itself. And this is probably the stage which you need to know the most about, because it involves a certain type of trial, a blind trial or a double-blind trial. So in these trials, they use these fake drugs, okay? So they have the drug they're testing, and they also have a fake drug. The fake drug is called a placebo. It looks just like the real drug, but it does nothing at all. So they get these ill patients. So let's say they get 100 ill patients. They split them into 2. You got 50 and 50. You give one half the real drug. You give the other half the placebo, but you don't tell them which one they're getting. And then you monitor what happens. Is the drug helping? Is it working? Are they feeling ill? Anything like that. And the reason they do that is to prevent the placebo effect, because sometimes people take drugs and they think they feel better. They think it's improving their health, but it's actually not, because subconsciously they're influenced. They're like, oh, I just took this drug. It must be working. Yeah, it is working. So it prevents that. That's a blind trial. But you also have double-blind trial. So double-blind trial is when they give you your placebo or your drug, the patients don't know which one they're receiving, but at the same time, the doctors who are analyzing the results don't know either. They only find out right at the end. Because again, think about what the doctors need to do. They need to analyze all this data. They need to decide whether this drug is working. But if they don't know, if they think they've been given the real drug, they might be influenced subconsciously, not intentionally. They might think that it's working. They might be looking at a graph and they'll be like, oh yeah, it's going up, so it means it's working. But if they don't know, it reduces that chance of a placebo effect. So it's very important for you to be able to explain that effect in your exams. Once all of that's done, the company who made the drug is basically saying, yep, this is ready to use. We can sell this out. But the government's not going to let you sell that. You need to do peer review. It needs to be tested by someone who's not part of that company. That company wants to make money. So it might be bias. In the validity of the whole trial needs to be checked by an external scientist who checks all the data to see that it's valid. Only then, once it's done all of these 5 steps, only then can this drug be released to the public. Placebo, doctor doesn't know. Sometimes they know, sometimes they don't. Yes, placebo effect. So we look for those 3 things when testing on animals: the toxicity, dosage, and efficacy. Yes, exactly right. Exactly right. All right. Nice. Next session, photosynthesis and respiration shouldn't take too long. So combined, guys, I'm going to let you guys go now. Triple, guys, we just need to go through monoclonal antibodies and plant defenses. I'm going to try and get that done now. I'm going to make the break And because it might take a bit longer, I'm going to make the break until, because I think the last session will be a bit shorter. Let's make it, I do 20 minutes, 2:30. Let's say 2:35, guys. Come back at 2:35. Combined, guys, you guys can go and come back at 2:35. Triple, guys, let's go through monoclonal antibodies. So monoclonal antibodies has the word clonal in it. They're clones of things that make antibodies. So clones of antibodies and the lymphocytes that make them. So remember, lymphocytes, they're white blood cells that create antibodies. So if we can copy lymphocytes, we can rapidly produce clones of antibodies, and we can use them for several different things. All right. You need to know the process of creating monoclonal antibodies. So what you do, you get a poor mouse and you inject it with a pathogen, some sort of illness. What's that going to do? It's going to trigger an immune response. They're going to fight this pathogen, right? So they're going to produce all these antibodies to try and fight them. Now, what we do is we extract those antibodies because we know those antibodies are perfect for this pathogen. We extract those antibodies and we fuse them with a cell which rapidly reproduces. Tumor cells. Do you guys remember? Tumors have cells in them which rapidly reproduce, right? So if you fuse a lymphocyte with a tumor cell, you've basically got this hybrid cell, a combined cell. We call that a hybridoma. A hybridoma is just this lymphocyte which rapidly reproduces, which means that all these antibodies on it are also going to rapidly reproduce, divides rapidly, and you've basically got clones of the hybridoma, and the antibodies on them are what we call monoclonal antibodies. All right. So we can design these monoclonal antibodies to be specific to specific antigens because we control what we put inside the mouse, right? So if we know what antigen is going inside the mouse, we know what type of antibody we're going to create at the end. So using this, we can use that for things such as pregnancy tests. So pregnancy tests obviously are there to tell a person whether they're pregnant or not. And they work by urinating on this area here. It's kind of like chromatography. The liquid rises up and up and up and up, and you have these different windows where you can see different things. Okay. Now, this is a bit confusing, but in the pregnancy test, you have mobile antibodies, which are bound to something called HCG, right? So they bind to HCG. So HCG is a hormone which is only produced by pregnant women. So if you're pregnant, in your urine, you're going to have this hormone called HCG. And this antibody is designed to bind to the HCG. So that mobile antibody, mobile means it's free to move. It's free to move up. It's also bound to a blue dye. That blue dye is what's going to be responsible for giving you those lines. If you guys ever done a COVID test, you guys see that you see lines, right? We've got a control line, and then we've got a test line, the results window. If you get 2 lines, it's a positive test. If you've got 1 line, it's a negative test. So the control window, let me explain the results window first. So in the reaction zone, you've got the mobile antibody, which is free to move. In the results window, you have immobilized. This means they can't move. So can't move. They're stuck to the pregnancy test, but it binds to HCG. That's the hormone. So it'll only bind if the woman has HCG in her urine. Then on the control window, you also have immobilized antibodies, but these are different antibodies. Can you see they're green? They only bind to this first mobile antibody. Remember, the mobile antibody is moving up, 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 up. So once they reach the control window, all of these mobile antibodies will attach onto the control window antibodies. And that's why you should always get a blue line up here. You should always get a blue line there because they should always attach onto the mobile antibody. If they don't, then that means your test is broken. So that's why if you never see a control line, even on a COVID test, it means you have to throw that test away. It means it hasn't worked properly because it has to always bind to the antibody. So if a woman is pregnant, she has HCG in her urine. This is this pink thing here. So as this mobile antibody moves up, it will reach the first results window. And remember here, the antibody binds to HCG. So it will stick the mobile antibody onto the immobilized antibody. And you're basically going to get a blue stripe here because all of these antibodies with HCG on them are also going to bind to the results window because they bind to HCG. So you'll get a line there. At the same time, you also get a line at the top because some of them are just going to carry on going up. Some of them might not have HCG on them, but they'll still bind to the mobile antibody and produce a blue line here. So if you've got 2 lines, it means you're pregnant. If you only have 1 line, that's because you don't have any HCG in your urine. So it won't bind to the immobilized antibodies in the result window. It'll carry on going up and you'll only get a line in the control window. It's very confusing, but I'm hoping once you go back and listen to what I said again and slow it down, if you still don't understand it, it'll make sense. Do you have to purify it? Yes, you do, but you don't need to mention that in an exam question. Is it a mouse that's always used? Not necessarily, but that's the one that always comes up in an exam. Question by GCSE study account. Let me see. You're answering someone's question, aren't you? Where is the immobilized antibody from? We create it using this process. These are the monoclonal antibodies that we're creating for. Yeah, you hate this topic. You know what? I think it's my least favorite as well. In biology. The other thing we use it for, monoclonal antibodies, is for treating and diagnosing cancer because cancer cells have these unique markers on the outside. They're kind of like antigens, we call them tumor markers. So you can create monoclonal antibodies which attach onto them. So we can create ones which attach onto them with a specific shape. And we can attach whatever we want onto these monoclonal antibodies. So like in the pregnancy test, we attach blue dye, right? We might want to attach like some sort of fluorescent dye so that we can identify where someone's got cancer. So you can basically monitor their body and see where they have cancer cells. Or we could attach a drug to it, which kills the cell. This way, it's going to attach to only cancerous cells, and the drug is going to be administered to that cancer cell and kill it. So it should kill the cancer cells without killing any healthy cells. So that's another use of it, diagnosing and treating cancer. B lymphocytes, yes, they're B lymphocytes, the ones that you use for monoclonal antibodies. That's correct. All right, last thing, very quick, this one. Plant diseases. Doesn't get asked much. How can you tell a plant is diseased? Well, might not grow as high. There might be spots on the leaves, especially for rose black spot. It might be rotting, abnormal lumps, malformed leaves or stem might be a weird shape, different colors, and there might be loads of pests around. If you have any of those things around a plant, it generally indicates that it's got a disease. You can identify the disease by, it's a bit general knowledge. You can read a manual or you can just look it up online. You can take it to a lab to be tested on, or you can test it for monoclonal, using monoclonal antibodies. Just like the pregnancy test, you can test for diseases as well. 2 ions you need to know about for triple, guys. Nitrates and magnesium. Both of these ions are crucial for plants for different ways. So nitrate ions are used for protein synthesis because we need nitrates to form amino acids, which help to form proteins, and that allows plants to grow. Magnesium ions we need to make chlorophyll. So if we have a lot of magnesium in a plant, it allows it to photosynthesize better. But if there's a lack of chlorophyll, it leads to chlorosis, and that's when you've got yellow leaves. If you have a lack of nitrates, it's stunted growth. The plant won't grow as much. The way plants defend against disease, there's only 3 that you need to know. Physical defenses, so the cell wall, remember the cellulose cell wall, that's a good barrier against pathogens. We've got waxy cuticle, layers of dead cell as well, like a tree. The bark of a tree is basically dead cells. It protects it. We've got chemical defenses. So some of them produce chemicals to kill bacteria or poisons to stop animals from trying to eat them. And then there's some with thorns and hairs, or the ones that droop their leaves to stop animals from eating them. Okay. Right, triple guys. That's all the triple content for Biopaper 1 done. Okay, so now it's just combined stuff. It's just photosynthesis of respiration. And I think we'll get through that quite quickly. So you can go have a break. We've got 15-minute break now until 2:35. So go and stretch your legs, have another snack maybe. Last 55 minutes or so. You guys are doing really well, smashing it. Could you attach sources of non-ionizing radiation to see where the cancer cells are? Yeah, you could, technically, you could. Yellow patch, yes, that would work. How does chemotherapy work? You don't need to know for your GCSE, don't worry. Yes, exactly, GCSE study account, that's right. What's the difference between physical and mechanical defenses? Physical are physical, like barriers. Mechanical are things which are like hurting anything that's trying to damage it. Yeah, study buddy. You're right there. That's good to hear, Blue G8. Good to hear. Thank you, guys. I really like the positive feedback. That's great to hear. Thank you. All right. Have a good break, guys. I'll see you in 15 minutes.
Welcome back, guys. Last stretch, guys. We're nearly there. We're nearly there. How's everyone finding it? How is everyone feeling? Are we good? Are we good? Are we surviving? Well done to all of you who stuck around. I know it's a lot of information. Just trying to go through everything you need to know for your GCSEs, trying to get you guys prepared as much as possible. I hope this has been useful to you guys. Yeah. Content's not the problem, it's the questions that's the problem, is it? That's great to hear. Thanks, Lexi. Welcome back indeed. Guys, we're nearly at the end. This one shouldn't take long. Thankfully, biopaper 1 last topic is, in my opinion, the easiest: photosynthesis and respiration. So before the end, I'm going to ask you guys to just fill out a review for us, because that helps us a lot. If you guys could do that, that would be so helpful. I'll put it on the chat at the end, because yeah, it really does help us out. Yes, that's good to hear. That's good to hear. You made loads of flashcards. Amazing, Ruby. That's good to hear. Okay, guys. So final one, bioenergetics. Easiest, in my opinion, maybe not for you. Also, the most important is this one, because this can come up in paper 2 as well. Bioenergetics is all about respiration and photosynthesis. You can get questions on both of those in your paper 2 as well. So please, if there's one topic you need to really know, is this one. So do you need to know paper 2 material for combined science? Yes, you do. You get 2 papers for every science. So you need to know all the paper 2 content. It'll be the same setup next week when we do bio paper 2 as well. Right, so I'll answer questions at the end, guys. I'm hoping this doesn't take the full hour. It shouldn't do. It's not that much, but once we've done that, we'll spin the wheel, see who is going to win the cram class prize and the paper 2 prize. One of you is going to win that. And then I'll answer any questions that you guys have. So let's start off with photosynthesis. We have explained photosynthesis already. It's the process where plants make their own glucose. That's their food. We can't make our own food. Animals can't make their own food. Plants can. That's what makes them different. So they get carbon dioxide from the air and water, and they use energy from the sun, sunlight, to convert that into glucose, which is their useful food, and oxygen as well, which is a byproduct. It happens in the chloroplast. Remember, in the chloroplast, there's chlorophyll, which is a green pigment, which absorbs sunlight, light energy. This process we call an endothermic reaction. So this is something we'll cover in chemistry as well. Endothermic just means it absorbs energy from the surroundings. So it's absorbing sunlight in order for this reaction to work. So what do you need to know about this? The word equation: carbon dioxide plus water goes to glucose plus oxygen, and the symbol equation. I know you might be thinking equation should be chemistry, but they do come up in biology as well. Just be careful here. Glucose is C6H12O6, and it needs to be balanced as well. Best way to remember that is just put 6s on everything except glucose. Okay, so CO2, 6 CO2 plus 6 H2O goes to form C6H12O6 plus 6O2. Another thing you need to know is how plants use their glucose. So that glucose they've made, that food that they've made, how do they use it? We've got a memory trick for that as well. We use scarf. So these are all the things that they can do with the glucose. So the first thing is they can convert it into starch. Starch is just like their stored form of glucose. Glucose is there to provide them with energy. If they don't need all that energy straight away, they can just safely store it in starch, in their leaves, in their roots, in their stems, and they can use it at a later time when they need it. Second one in scarf, the C, is for cellulose. Cellulose is a substance that you find in cell walls. We talked about this in topic 1. It protects the cells, the plant cells. Then we've got the A, which is for amino acids. Amino acids make up proteins.
Plants have to make their own amino acids. So we eat food to get amino acids. Plants just make it themselves. They basically get glucose that they've made from photosynthesis, and they combine it with nitrate ions. Nitrate ions they get from the soil through, remember, mineral ions? Mineral ions, sorry, they go through the roots. They absorb that through the roots, and then they combine it with glucose to make amino acids. The amino acids then combine to form proteins, and the proteins help the plant to grow. Then probably the main one is respiration, which we're going to cover in a second. Remember, respiration is how they release energy, transfer energy. Don't say make energy. You will get marked down if you say makes energy. Energy cannot be created or destroyed. And the last one, F, is for fats. They store some of the glucose, they convert it into fats and store it in their seeds to use it for another time as well. Scarf, yes, indeed, indeed. Well done, Dylan. All right. Photosynthesis can be sped up using different factors. We've got a memory trick here, and I actually think it's better for you to watch this video. So scan the QR code once this session's over, if you want to watch this video. It explains it better because I've got some animations here, but Likert helps you to remember the factors that affect it, but it also helps you to remember how it affects it. Li is for light intensity. So if you have more light intensity, more light, photosynthesis is going to be faster. That makes sense because you need that for energy, because it's an endothermic process, right? CO is for CO2. Imagine there's a small 2 there for CO2 concentration. They need CO2 concentration. They need a high CO2 concentration to photosynthesize as well, because that's one of the reactants for photosynthesis. And the T is for temperature. They do also need a high chlorophyll concentration. I don't have that in this memory trick because you don't need to know the graph for it. For all the other three, you need to know the graph for it. But the fourth one is chlorophyll concentration. The more chlorophyll they have, the more they're going to photosynthesize. So I'm just going to add that here as well. Chlorophyll concentration. How do they affect it? So you need to memorize these graphs, guys. So for light intensity, before we go through those graphs, actually, you need to understand something called limiting factors. Limiting factors. So all these factors I'm going to list that I've already listed out to you, they can sometimes be limiting. Limiting factor basically is a factor that needs to increase in order for you to increase the rate of photosynthesis. So, for example, on a warm night, the sun's gone, the temperature is good, the CO2 concentration is good, the limiting factor would be light intensity. So you basically need a higher light intensity. You need that to increase your photosynthesis. Therefore, light intensity is limiting it. On a cold day, let's say it's a sunny day, it's winter, it's midday, it's very sunny, so the plant's getting a lot of light, it's getting good CO2. The only thing that's limiting it is temperature. So temperature would be the limiting factor. And if you have a warm day in summer and it's midday, good temperature, good CO2 concentration, no, not good CO2 concentration, normal CO2 concentration, good light intensity, everything else is good. CO2 concentration is what's limiting it, because that needs to be increased to make the photosynthesis greater. So limiting factor is just a factor that you need to increase to make photosynthesis occur faster. And this is the graph for light intensity. So as you increase light intensity, the rate of photosynthesis increases, but it doesn't just keep increasing. It only does that up to a point. It levels off eventually. It goes flat. There's a plateau. That's because around this point, there's a limiting factor. And that limiting factor is stopping it from increasing its rate. And that limiting factor is always one of the other two factors. So light intensity is high enough. Therefore, what's limiting it? It's either temperature or CO2 concentration. We don't know which one it is, but it's one of them. Either one of those need to be increased to make this graph carry on increasing. Okay. And we've got a similar graph for CO2 concentration. As you increase CO2 concentration, the rate of photosynthesis also increases, but only up to a certain point. It levels off. This time, what do you guys think the limiting factor is? Let me know in the chat. What's the limiting factor here for when the CO2 concentration graph levels off, based on what I said? Do you need to know the graphs? Yes, you do. You need to be able to draw these graphs. You need to be able to sketch them. Then it says temperature. It could be temperature or light intensity. Exactly. Well done, guys. It could be one of the other two. So this rule works for these two graphs. The third graph is different, though. The third graph is for temperature, and you can see it's very different. It goes up and then it goes down. It goes down. Where have we seen this graph before, guys? Let me know in the chat. We've seen it earlier today. This exact same shape graph. No tangent needed here. No tangent. There we go. 5's R6, sorry, has said it. Is enzymes. There we go. Oh, look at this. Everyone's getting it right. That's showing me that you guys have been concentrating and you've learned something from this session, which makes me happy. So yes, this looks just like the enzyme graph, and it's explained the same. As temperature increases, there's more collisions between the enzymes, the substrates in the plant. Photosynthesis occurs faster up to an optimum temperature. That's when it's the fastest. After that, the enzymes start to denature. So remember, that's when the shape of the active site changes. The substrate no longer fits inside, and that causes the rate to go way, way, way down. That temperature is around 45 degrees. Okay, so just remember this graph is exactly the same for CO2. For temperature, it's the enzyme graph. There's no limiting factors to explain in the temperature graph. It's just, you're explaining it just like enzymes. Okay? No longer complementary. Exactly. Frequent successful collisions. I'm loving that. That's a bit of chemistry. So what questions can you get on this? Loads of questions are about greenhouses. How are greenhouses helpful for maximizing the growth of plants? So greenhouses are used by farmers to create products for humans using plants, right? So we have artificial things that help a plant grow better. We've got heaters. The greenhouse, where's heaters? Heaters here. The greenhouse is made out of glass, so it maximizes how much sunlight goes to the plants. But at the same time, they can also use artificial light. So at nighttime, when the sun goes down, they can use artificial light to maximize photosynthesis, and heaters to increase the temperature to make it the perfect optimum temperature. Not only that, they can use something called paraffin heaters. These are special heaters that give off carbon dioxide as well, because if there's more carbon dioxide, that also increases the rate of photosynthesis. Okay, so these are different things that farmers would do, but they have to be careful. They shouldn't spend too much money on these things, right? Because light heaters, they cost money to run. So they need to find like a limit to how much light to give, how much heat to add to the greenhouse. And they do that by figuring out their costs, because they're creating this product and they're selling it and they're getting money out of it, right? So they need to make sure that the extra money they're using to run the lights and the heaters are still profitable when they sell the plants. They're going to sell more plants, but is it worth it? So the questions that you get on this are data questions, where they give you data and they ask you why would a farmer not use a light intensity that's really, really high? They'll give you like a really high example, and then you look at the data and you'll see, well, because the rate of photosynthesis is already very high at a certain number, it doesn't need to be increased anymore, because it minimizes the extra rate that you get from that. it's data questions. It's application questions. So you need to use that data and your own knowledge to answer those questions. Can you mention amount of chlorophyll as a limiting factor? Good question. You can, but I'll try and avoid it. You usually talk about these three in these questions. You saw this on a combined paper. Yes, photosynthesis and respiration comes up a lot. So with this, because it's such an important topic, you've seen a question on this where you can't mention costs. Interesting. So it might be a very specific question where they've already talked about costs. So when they say you're not allowed to mention something, it usually means that they've already mentioned it in a question, so you're just repeating what they're saying, or they've told you something else which is guiding you towards a different answer. So required practical, this is the pondweed one. People got confused between this and the transpiration experiment. Remember the transpiration one? That's the one with the water reservoir, the potometer. This is the pondweed example. So here you have pondweed, which is like a plant which grows in water, and it produces oxygen in the form of bubbles. And what you can do is you can test how fast photosynthesis is occurring by counting how many bubbles there are. The more bubbles the plant produces, the faster the rate of photosynthesis. So if we get a light source and we shine it, I don't know, let's say 10 centimeters, we'll get a certain amount of bubbles after a certain amount of period. But then when you bring the light closer to 5 centimeters, let's say, you get even more bubbles produced. That tells you that the higher the light intensity, because the light is closer, the faster the rate of photosynthesis, because more oxygen is being produced. The more accurate way, rather than counting bubbles, because it's quite tricky to count bubbles, they're really small, the more accurate way of doing this is by using a gas syringe. So the gas syringe basically collects the gas. Rather than you counting the bubbles, it literally collects the gas, and you can read off how much gas has been collected, the volume of gas. And what you can do is you can keep repeating this experiment at different distances. And you're changing one variable. So you can either change light intensity or temperature. Those are the two that gets asked in exams. Some room for error here. These light sources, sometimes they get hot. So they might ask you, what could you do to improve your experiment? Instead of using a filament lamp, you can use an LED. An LED light does not get hot, because you don't want these lamps to get hot, because otherwise that's going to also affect the photosynthesis rate, right? So you can switch it out for an LED light. Why does a higher light intensity mean a higher rate of photosynthesis? Because that's what provides the energy for photosynthesis. So if you have more light, it's going to provide more energy, so photosynthesis can occur faster. What if resolution of syringe is too small? Yeah, then I don't think that's going to come up in the exam. I don't think it can be too small. I think we'll be fine. When we buy a syringe with a better resolution, repeat. Yeah. Is photosynthesis controlled by enzymes? Yes, it is. Yes, it is. Inverse square law. Someone says they hate this. It's a rule that basically tells you what happens to light intensity as you increase the distance of a lamp. So if you have a lamp further away from you, the light that hits you, it's less. So imagine someone shines a light right in your face. It's going to be very, very bright, right? The light intensity is going to be incredibly high. And then they move away 5 meters. It's going to go down, the light intensity. Then they move away 10 meters. It's going to go down even less. But the relationship isn't, because they've doubled their distance, light intensity has halved. That's not the relationship. Loads of people think that. They think, okay, this person's 5 meters. I'm getting this much light. They've now moved 10 meters. They've doubled their distance. So I should be getting half the amount of light. That's not the relationship. The relationship is the inverse square law. That's the relationship that it follows. That means If you double the distance, the light intensity goes down by 1 over the distance squared. So look here, we've got a lamp at 5 centimeters, and that produces a light intensity of 16 AU, arbitrary units. Yeah, that's not that important. Now we move that lamp to 10 centimeters. What we've done there is we've doubled the distance. We've multiplied that by 2. So people will automatically think that means this should go down to 8 AU. Because we've doubled the distance, light intensity halves. That's not what you do. Inverse square law means you do half squared. Inverse square law means you do half squared. So you do 1 over 2 squared, which is basically 1 over 4. This quarters. So you do a quarter of 16 and you get 4 AU. That's basically what it means. So look here, we've got an example here where you want to work out light intensity. We've got a distance of 5 centimeters and we want to work out the light intensity. You do 1 over 5 squared, 1 over the distance squared. Then we do 1 over the distance squared again, and we keep doing that. You just need to remember to square the bottom of the fraction and you get the answer here. Okay, it seems complicated, guys, but it's really not. You just need to memorize this 1 over the distance squared and just put it into the equation. Okay, that's good to hear. SKZ, thank you. Why do we measure the distance of the gas syringe from the capillary tube? Okay, so that's a good question as well. This is sometimes asked. I haven't seen it asked much, but sometimes if you have a bubble here, you can see how fast that bubble moves. And from that, sorry, not how fast the bubble moves, how big the bubble gets. You can measure the size of the bubble. The bigger the bubble is, the more oxygen that gets produced. Okay, and I think that's what we say here in the notes. So there's different ways to do this. You just need to see what comes up in the exam and relate it to whatever the question's asking. Is there a possibility of question that will require us to find the distance using light intensity? Possible, could be possible. That's not. I don't think they're not allowed to do that. I think they're very much allowed to ask you that, but I've never seen that. If the question says you must have calculation and describe, what would you say? You'll do these calculations here. And then you compare these numbers. You'd say 0.01 is 4 times smaller than 0.04, and then that proves the relationship. Do we need to control the light in the photosynthesis required practical? The color of light, yes, that's one of the control variables. Temperature is one. If you're investigating light intensity, temperature is one. Color of light, you'd need to control that because that could affect how much light is absorbed as well. You have a feeling this could come up on exams? We don't know. Could do. Instead of made, you would say produced. Fine. That's fine. I'm not an examiner myself, no. But I've been teaching for over 10 years. How does color affect it? You don't need to know, but I'll tell you, different colored lights have different wavelengths. And chlorophyll only absorbs certain wavelengths of light. Okay, last thing is respiration and exercise, guys. So respiration is the process where we release energy, transfer energy, not produce energy, by breaking down glucose. Okay, this is the opposite of photosynthesis. It's exothermic. It's not endothermic. It's exothermic. So that means it releases energy, which that is literally the definition, right? We're getting glucose and releasing energy. We use it for 3 things, mainly keeping warm. We remember our body temperature is 37 degrees. It's never 37 degrees outside in the environment. So we always need to physically heat ourselves up so that we're hotter than our surroundings. That takes a lot of energy. People don't realize, even when you're sleeping, you're using a lot of your energy just to keep warm. Contracting for movement, this is the one that people probably think of the most when they think of energy. For moving, you need energy, yes. For walking, for doing anything. Even for breathing, because you need your diaphragm to move. And also for reactions, when we want to build larger molecules. So amino acids being converted into proteins, for example, that needs energy. 2 types of energy you guys need to know about: aerobic, anaerobic respiration. Aerobic. Think of air. What is the air made up? What do you think of when you think of air? You think of oxygen. Aerobic respiration is respiration with oxygen. It's the most efficient way. It's the most ideal way for us to respire. It releases the most amount of energy from the glucose. This is what we always want to do. If it was ideal, we would do that. Okay? We're always doing it. And this is the one that occurs in the mitochondria. Anaerobic respiration is the opposite. It doesn't use oxygen. Oh, sorry. And also the equation for respiration, easy, because if you know photosynthesis, you know aerobic respiration. It's literally photosynthesis flipped around. Easy way to remember that. So glucose plus oxygen goes to carbon dioxide plus water, and even the simple equation is just flipped around. Okay. Anaerobic respiration is respiration without oxygen. So that happens when you don't get enough oxygen. So when you're exercising, for example, you need more oxygen to keep you running. We'll talk about that when we do exercise. It doesn't produce as much energy. It's not as efficient because we say the oxidation of glucose is incomplete. Okay. When this happens in humans, a different reaction happens when compared to plants. So don't get these confused. When anaerobic respiration happens in humans, it's just glucose getting converted into lactic acid. When it happens in plants and yeast, the glucose gets converted into two things, ethanol and carbon dioxide. So we use this to make things. We use carbon dioxide in bread making. For bread making, you get dough, you add yeast in the dough, and then you put it in the oven and the bread rises. If you cut up bread, it's got loads of air gaps in it. It's very spongy, right? That's because carbon dioxide has been released by the yeast. The yeast is respiring. The yeast is a type of fungus. It's a living thing. We also use it to make alcoholic drinks. The ethanol that's produced makes a drink alcoholic. So if you add ethanol to a drink, it basically turns into an alcoholic drink. Where are these questions? Exactly, Blue G8. That is correct as well. That is correct. Bread and beer. Yes. All right. Are we ready for the final one? I think we're going to finish early today, which is good because we started off not. We started off late, but I think we've caught up really well, guys. But yeah, well done, you guys. Metabolism and exercise. So metabolism is all the chemical reactions that occur in your body. Okay, it's the sum of all the chemical reactions that occur in your body. Loads of people think it's to do with food and how fast your food is digested in your body. That's part of it. It's not the complete picture. It's actually all the reactions that happen, including that as well. So they're controlled by enzymes. These reactions are controlled by enzymes. And you need to know 5 different examples. These are all in the spec, so I've listed them out. So let me go through them. One reaction that we carry out in our body, a metabolic reaction, is glucose being converted into glycogen. So do you remember I said plants store their glucose as starch? We don't store glucose as starch. We store it as glycogen. We store that in our liver and then in our muscles. We're going to cover that in paper 2 next week. That's a paper 2 topic. But plants might use their glucose to convert it into starch and cellulose, sorry. We've mentioned that already. The second reaction is making lipids. Remember I said when you break down a lipid, you get glycerol and fatty acids. We sometimes need to make lipids. So we do the opposite. We get 3 fatty acid molecules and glycerol. We join them together, and then it makes a lipid. It makes a fat. Sometimes we need to make amino acids, and I've told you this already. To make amino acids, we combine glucose with nitrate ions, and that makes proteins. That's how we make proteins. That's how we grow. These 3 at the top are all us making things, but we also have metabolic reactions, which break down things. So respiration is a metabolic reaction. We've just covered that. So that's releasing energy. And also breaking down proteins. We learned this in paper 2 as well. Proteins can be broken down to form urea, which basically forms in your urine and you excrete it. So 5 examples you guys need to know for metabolism. Yes, there is a mark scheme for the exam questions. We'll send it all after. I think you guys should have got the physics one right. Do you guys get the physics mark schemes for Sunday's session? Is there a way to memorize the metabolic example? Not from me, unless anyone else knows one. I've tried to come up with one. Some of these are really hard to come up with a memory trick for. Yes, you're saying yes, 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 yes. Okay, that's good to hear. Why can we store glycogen? Yeah, basically, it's easier for us to store. We can't store glucose. Having high levels of glucose in our blood is dangerous. That's how you get diabetes. A wise pharmacist mixes drug fresh. Ah, I like that. That's for aspirin, willow, penicillin, mold, digitalis, foxgloves. I like that. GCSE study account. That's a way to memorize that. Nice. A wise pharmacist mixes drugs fresh. I might make a video about that. Why is the example different on the light intensity page, different to the table? Okay, I'll come back to that. Ask me again at the end, Sammy. What is glucagon? That's a paper 2 topic. We'll go through that in paper 2. It's a hormone. Is it a good idea to make flashcards? Absolutely. Now's the time to make flashcards. After this, you're not going to have much time to make flashcards, so make as many flashcards as you can in Easter, yeah. You don't get why starch is insoluble. You don't need to know why. Just know that it's insoluble. It's just a chemical thing. It's something to do with chemistry. You don't need to know that for your GCSEs. Don't worry. Okay, guys, I'll answer these questions at the end. We're nearly done. So we're going to finish this off. We're going to spin the wheel, and then I'm going to answer these questions now. We're really good for time. You guys have been smashing it. Thanks for keeping up on the chat as well. It's really good participation for you guys. Last final thing, guys, with there, end of paper 1 biology exercise. What happens when you exercise? So imagine you're doing a light jog, okay? What's happening when you're doing a light jog? Your muscles are contracting. They're moving, contracting, relaxing, more than usual. So you need more energy to do that. Where do you get your energy from? Respiration. What's our ideal form of respiration? Aerobic respiration. So that's why we need more oxygen, because aerobic respiration is respiration with air. So we start breathing a bit more heavily. You're doing a light jog, a bit more heavily. Your breathing rate increases. Where's my highlighter? Your breathing rate increases. Your breath volume increases. So you breathe heavier and faster, and your heart rate starts to increase slightly. Okay. And that should provide you with enough energy to do a light jog, because you're giving your body enough oxygen. Now, let's say you start sprinting. When you're sprinting, something different happens. Because a sprint is so vigorous, your body respiring aerobically doesn't give you enough energy to sprint. It's not fast enough. So in order to sprint, we need to anaerobically respire. Aerobic respiration is not happening fast enough. We're not getting enough oxygen for aerobic respiration, even though we're breathing really heavily, really hard, high heart rate. It's still not enough for us to sprint. So we start to respire anaerobically without oxygen. We're like, okay, listen, it's not coming fast enough. We're just going to break down this glucose and form lactic acid. See, this is where the problem arises. Yes, it allows us to move faster. So it's useful for like a race or when you're trying to run away from something. But it produces lactic acid. Lactic acid causes fatigue in our muscles. It gets our muscles tired. It starts to build up in our muscles, and they start contracting less efficiently. So this is why you start getting tired really quickly when you're doing vigorous exercise. And you get cramp. That's cramp. Lactic acid building up in your muscles is literally... that's the definition of cramp. So imagine you've just done a 100-meter sprint. you've run as fast as you can, and you finished your sprint. What you'll notice, as soon as that sprint's finished, you're still breathing really heavily and really deeply. Your heart rate is still pumping fast. Even though you finished the race, even though you're not moving, you drop to the floor, you're still breathing really heavily for like a minute or two, okay? The reason for that is because that lactic acid needs to be removed now. You've basically borrowed oxygen. We call it oxygen debt. You should have had oxygen, but you weren't getting fast enough, so you're kind of borrowing it. So we call that oxygen debt. And what you're doing is you're paying back the oxygen debt. And you use that oxygen to remove all that lactic acid that's built up. So oxygen reacts with the lactic acid, and it goes to the liver, and it gets converted back into glucose again. It gets removed, sorry. It gets removed. So that's why after a very vigorous exercise, you're still breathing heavily, even though you're not exercising anymore. So that's oxygen debt. You're basically removing the lactic acid. Okay, oxygen debt is triple? No, it's not. It is not. Everyone needs to know about oxygen debt. It is something that you need to know. It's higher, so it's not for foundation, but it is for combined and for triple. Okay. Actually, even this section here, that's not even higher. That's for everyone. Everyone needs to know this, guys. Okay. So make sure you know it. All right, guys, that's everything in biology paper 1. Congrats. Thank you for bearing with me since 10 a.m. You guys smashed it. 75 of you still in here. Well done, guys. So like I said, I'm going to do the spinning the wheel in a second. I'm just going to drop the review, just a chance for you guys to review. So please, guys, let us know. Would love to hear your feedback. Thank you for all of those of you who gave us some really good reviews on the last session. It really does help us to just keep things running. I'll just drop it in the chat now. Thank you, guys. That's really good to hear. That's what I like to hear, that it's been helpful to you guys and it'll help you for your exams. Yes, please, 5 stars would be very useful. Thank you very much, GCSE account. Right, are we ready for the wheel spin? We finished early, guys, so I'll stay behind after to answer some questions. 6 stars if possible. Love that, love that. Yeah, I'll answer that question. Sammy, yes, one more time. I'll ask you after the wheel spin, okay? Ask again. Right, let's go for the wheel spin. Wheel spin, wheel spin. Let's get the wheel. Wow, there's a lot of names here. Let me get it up on the screen. There we go. Drum roll, guys. One of you, whoever gets this, whoever it lands on. The paper 2 sessions next week free, and the cram classes that are happening the day before your exams completely free. It was Dylan last time. Let's see who it is. Get ready. Let's spin. Drum roll, drum roll, drum roll. It's going to land on Mercy. Congratulations. We'll send you an email. You're getting all of that for free. You smashed it. I don't know if you're in the chat. If you're in the chat, say hi. Congratulations. You're going to get all of that for free. And yeah, we'll send that to you by email. You were so close. Oh, you got it. There you are. Mercy, well done. Well done. Smashed it. We'll do one more in the chemistry one as well, guys. I'll do one more, but yeah, we'll send you an email. Well done. And I'll stay behind now, guys, to answer some questions. So those of you who I'm not going to see again, take care. Good luck with everything. I'm doing this again on Thursday. Chemistry. Chemistry is a big one, guys. It's a lot more content. It's 5 topics this time. Physics and biology had 4 topics. It's going to be 5 topics on Thursday. It's going to be full on. Triple people are probably going to get even less of a break. But it's a good one. So I hope to see you guys there on Thursday. If you guys don't have any questions, you guys can shoot off. I'm just going to be answering some questions. It might be useful to stay behind while I answer these questions. for your revision as well. Let's hear what you guys have. Any last minute questions on paper 1? How to revise science content. Go topic by topic. Right now, during Easter, go topic by topic. Go through the notes. Once you're happy with the notes on a topic, do questions on it. If there's any, and then at the end of it, go through the spec points, check that you know everything, and then move on to the next topic. If you don't know something, go back and revisit the notes again. Repeat, 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 repeat. You already covered the whole chem spec, but you'll still be here. Love to hear that. It's good. Even though you've covered stuff, it's good to hear it again. It helps to reinforce that. Remember, guys, when you listen to something, you retain something like 60, 70% of that of the information, maybe less. But if you cover it again, then you're more likely to keep it. The more times you recover the topics, the information, the more likely is that it's going to stay in your head. What would you say oxygen debt 2 marker? Oxygen debt 2 marker. Something like anaerobic respiration occurs, meaning no oxygen is being used. Oxygen is needed to remove the lactic acid that's built up during the anaerobic respiration, something like that. So when we get more oxygen to repay the oxygen debt, the oxygen reacts with the lactic acid to move it, or does it react with lactic acid and then send it back to the liver? It basically goes to the liver to be converted back into glucose. So that reaction happens into the liver. Yeah. You missed the first part of the live. Don't worry. The whole thing's been recorded. You can literally rewind right now if you want. Watch it from the start, print out the revision guides, and act as if you're there live. How do you answer 6 markers for full marks? It's a tricky one to say, because if you've ever seen a mark scheme of a 6 marker, they're always really different. And they're marked differently. It's not 1 mark per point. So sometimes you need to say more than 6 points. Sometimes you need to say less than 6 points. So best advice I can tell you is just answer the question as much as you can, and don't worry too much about how many points that you've made. That's the best thing to do there. But do as many as you can. I know people, even my students, my tutoring students, they always skip out the 6 markers when we're doing exam questions. But you need to practice them, guys. You need to practice them. Don't get, you know what the average for a 6 marker is in the whole nation? It's something like 2 to 3 out of 6. That's the average mark someone gets from a 6 marker. So the rest of the country are not, on average, are not going to get 6 marks. So if you can get a bit more than that, then you're already doing something better to get a better grade. Practice them, guys. Look at the mark schemes. It's so useful to look at mark schemes when you're revising. Lactic acid is broken down in the liver. Yes, it is. Could we just use the flashcards on the website, or do we need to make more? I say make a bit more, Sammy. Flashcards on the website, yes, you might not see all of them. There might be stuff that have been messed up. I'd say use the notes and make your own flashcards rather than copying the ones on the website, although the ones on the website are good. No worries. What's the most effective way to revise this time? Triple science, we have less time. You still have a lot of time. Remember, it's Easter. You have how many more days? 10 more days? 10 more days? 10 more full days, guys. You have the time. After Easter is when it gets tricky. Right now is the time for you guys to lock in and to do as much revision as you can, because the exams are going to come so fast after. So you do have time, trust me. Just go through the content one by one and try and cover as much as you can. Did you do past paper lives? Yeah, we will do that on FRR classes. Once we finish the content, I think we've got 1 or 2 more weeks of content, and after that, we're going to go through past paper questions. Minterplane, no, I'm not. Where can I find? you should have got it emailed. If you didn't, then email help@flashrevisionlab.com, and you'll find out where to do it. No worries. What did I get for my GCSEs? All A stars for my sciences, maths, and English. So you're in good hands. Don't worry. I also did. I got top marks in A levels as well. All A's. You couldn't get A stars back then. And I did chemical engineering at Imperial as well. So I know my stuff. Don't worry, guys. And I've been tutoring for over 10 years. Why is the example different online? Oh yes, Sammy, let me answer your question, family. So you said, why is the example different? Which one are you talking about? Are you talking about this and this? So this one is describing how light intensity gets changed with distances. This is when you don't get given a light intensity at the beginning. If they just give you a distance and ask you to work out light intensity, you'd use this. This one is when they do give you light intensity and they ask you to work out another light intensity. Does that answer your question? So the table is for if they just ask you to calculate it just using the distance. Yes. A stars. Do you cover any misconceptions students have made over the years? When we do questions, I do. Nice. Imperial and Oxford, yes. All good unis. Dennis, email again. Sorry. You were the last one, right? In the last one. It's probably because we've been so busy just organizing these boot camps. Send us an email again. You'll definitely get your prize, don't worry. You've got it before already. You know we'll give you a prize. All right, guys. We can do when we in the FRL classes, we'll go through some exam questions. So what we'll do is we'll send you guys the answers to these the exam questions in this revision guide. We'll either do that today or tomorrow, and you guys can try out the exam questions, and then at some point we'll do exam questions as well. Yeah, because I really wanted to just focus on the content today, especially biology. There's so much content. I just wanted to get you guys on that. I don't want to fry your brains too much. Can we do a Kahoot? Ooh, I was thinking of doing one tomorrow, but I don't think I'm going to. I think I'm going to wait until after Easter to restart the Kahoots, guys, on Wednesdays. So yeah. The wheel spin has happened. We've got our winner already. It was Mercy. Guys, thank you so much. It's been a pleasure. I'll see you hopefully on Thursday. Like I said, please drop a review. I love hearing the 5-star feedbacks. It's really good for us, and it helps us out a lot as well. Study hard. Go get some rest now. Maybe do a bit of revision after if you want. If not, give yourself a rest and start again tomorrow. Do as much as you can over this Easter. And I will see you all on Thursday. Looking forward to it. All right, guys. Bye.